The Amyloidosis Forum is a public-private partnership between the FDA and ARC. It was founded or established in 2019. The overarching goal of the forum is to optimize innovation and accelerate the advancements of new therapeutics by addressing scientific gaps and needs through the conduct of mutually beneficial scientific activities in the pre-competitive domain that aid the scientific evaluation of the efficacy and safety of new therapeutics for amyloidosis. The forum is an international initiative now involving regulatory agencies from Europe. We have EMA participating today, MHRA, and from Japan, PMDA. So I'm really grateful to see the expansion of the forum, as well as many international contributors from the scientific field. We've had a number of prior forums that meetings that we've had both at FDA and at workshops, and these have focused to date on, thankfully not me, on AL amyloidosis, and the last meeting we had on imaging. So it's a really exciting time today to be introducing the it's really, sorry, the slides are a little mixed up. So it's really exciting time to be introducing ATTR amyloidosis. The first generation of drugs approved in ATTR have totally reshaped the research environment. When we had our first meeting with FDA in 2015, there were no drugs approved, and patients really talked about the trauma of living with a disease that was progressive and there was no hope. The landscape has really shifted significantly, and now there are other challenges in how we design trials when people are living longer and we have treatments available. So really excited to be setting this forum meeting up. I do want to thank the Scientific Steering Committee who really do a lot of work behind the scenes to set this agenda, and we have an impressive agenda today. The meeting, the objectives are really to understand the impact of novel treatments and the unmet needs, to define new approaches to designing clinical trials, and outline regulatory approval and reimbursement challenges, and really define from this a forward-looking action plan. Today's agenda, and it is a packed one, so you should all have a copy of the agenda. If you are online, there is on the amyloidosisforum.org, one word, you'll be able to see the agenda. But it's really a meeting in two parts. The morning session will focus on updating the unmet needs for patients and from a patient and clinician perspective. And the afternoon session will focus on challenges facing drug development and access. So lastly, and my job will be complete, please, for speakers, state your name before starting to speak and introduce yourself briefly. Questions will be addressed within each moderated discussion and designated Q&A sessions. Please be mindful that this is not the appropriate venue to seek specific advice on a trial design or about an individual's health. There's a standing mic in here, and there also, you can ask questions at the Q&A box if you're online. We are committed to starting on time and ending on time. So with this, I'm going to hand over to Dr. Maurer, who is the co-chair for today.
So thanks, everyone, for coming. Good morning. Just a few comments. I'm very grateful to ARC, the FDA, and everyone here for attending this meeting. Colleagues who've come from far distances to share their thoughts and ideas. I'm also grateful for the National Institute of Aging, who's helping to support this meeting as we try to advance the science in developing new, effective, safe therapies. And I just remind you that the main reason we all do this is our patients. These are wonderful people who suffer from a terrible condition that's got better, but there's a whole host of unmet needs that are still out there. And so welcome a vigorous discussion today and everyone's thoughts in moving forward with that collective agenda. It's my pleasure to introduce my co-chair, Rosalind Adigan.
Thank you, Matt. Can you hear me? Can you hear me? Okay. Good morning. My name is Rosalind Adigan, and I am a medical officer within the Division of Cardiology and Nephrology Drug Products here at the FDA. And I echo Dr. Maurer and Ms. Lozada's sentiments when I say I want to welcome everyone for attending in person and for our virtual audience. It's my pleasure to welcome you to the FDA campus. I have no disclosures, but just to reiterate some of the comments earlier stated. The findings and conclusions in any of the presentations that you will be hearing and seeing today have not been formally disseminated by the FDA and should not be construed to represent any agency's determination of policy. The mention of commercial products, their sources, or their use in connection with materials reported here is not to be construed as either an actual implied endorsement of such products by the Department of Health and Human Services. So how did this public and private partnership come about? So the Critical Path Initiative, launched in 2004, is FDA's national strategy for transforming the way FDA-regulated medical products are developed, evaluated, and manufactured. Through collaborations with various stakeholders, these initiatives seek to further foster innovation and bring efficiency to the review process. One of these mechanisms is the public-private partnership. Through this, CDER works with stakeholders to address specific regulatory scientific gaps or needs that are beyond the capacity of a single stakeholder or a single organization. The purpose of the PPC, which have been stated, aims at bridging scientific gaps in drug discovery and development. We leverage expertise and resources for the conduct of mutually beneficial activities in the pre-competitive domain. We also promote development of research tools, platforms, clinical databases, methodologies, and predictive models to advance our knowledge of diseases and safety profiles of drugs. And is the ultimate goal that the project results generated by these public-private partnerships are made broadly available to benefit public health. So as we move to the program today, what are some of the general guiding principles? Can we advance this slide? And this is just a snapshot of the many public-private partnerships, and this is not an all-inclusive list. There is so much more with which CDER engages. So our participation today is limited to providing a general perspective on regulatory standards, scientific issues, and gaps related to pre-competitive drug development. Therefore, we cannot comment on specific regulatory applications, products, or other non-public information. We cannot provide specific opinions on quality and quantity of scientific evidence for regulatory decision-making. We are also unable to provide opinions on what conclusion an official regulatory review might reach based on scientific evidence, or provide recommendations or comments on submissions intended for the FDA review. We cannot also give specific advice on a proprietary drug development program. But anything else outside these, I believe, will be fair game for us to discuss today. So with that, I would turn it over to Dr. Maurer. Thank you.
Thanks. So our first session this morning is an overview of ATTR amyloidosis and treatment advances. I actually can't think of anyone better in the entire world to deliver this lecture than Julian Gilmore, a colleague from the National Amyloid Center across the pond, as we say. Julian, I hope you're there, and we look forward to your talk.
Can everybody hear me?
Very well.
And you can see my slides? Yeah.
Yep.
So good morning, everyone. Thank you, Matt, for the kind introduction. And I'd like to thank the Amyloidosis Forum and the ARC and the FDA for asking me to provide an overview of transthyretin, or ATTR amyloidosis, including the recent advances in its treatment. I'm sorry not to be with you in person. So amyloidosis is a disorder of protein folding in which so-called protein fibrils are deposited in between cells within tissues, causing disruption of tissue architecture and disturbance in organ function. Amyloids identified by pathognomonic green birefringence when a tissue biopsy is stained with Congo red and viewed under the microscope using cross-polarizing filters. There are in fact more than 30 circulating proteins that can misfold and form amyloid fibrils in humans, and they form the basis for the classification of amyloidosis. In other words, the amyloid fibril protein in AL amyloidosis is a monoclonal immunoglobulin light chain. In AA amyloidosis is the acute phase protein serum amyloid A protein. And in ATTR amyloidosis, the subject of today's discussion is transthyretin, or TTR for short. TTR protein in the circulation is entirely produced by the liver, and it exists as a tetramer. There are two pathways that are proposed to lead to misfolding of TTR into amyloid fibrils, the so-called dissociation pathway and the cleavage pathway. In the dissociation pathway, the TTR tetramer dissociates into monomers, which misfold, if you like, into amyloid fibrils. In the cleavage pathway, the TTR tetramer is specifically cleaved, and the cleavage fragments misfold into amyloid fibrils. So ATTR amyloidosis is a multisystem disease which affects the eyes, the nerves, connective tissue and joints, kidneys, GI tract, heart, and central nervous system. However, the predominant clinical manifestation at the time of diagnosis are due to, one, infiltration of the heart, resulting in arrhythmias or symptoms of heart failure. This predominates in non-hereditary or wild-type ATTR amyloidosis, but is also present in hereditary ATTR amyloidosis. Two, infiltration of the peripheral nerves, typically in hereditary ATTR amyloidosis, resulting in pain, loss of sensation, weakness, and gait disturbance. And three, infiltration of the autonomic nerves, also typically in hereditary ATTR amyloidosis, resulting in postural dizziness, GI disturbance, impotence, weight loss, and loss of mobility. In the absence of treatment, the accumulation of amyloid deposits in these organs leads to a gradual decline in quality of life, which culminates in end-stage heart failure, and in the case of hereditary ATTR amyloidosis, inability to walk or even stand, constant pain, and incontinence of both urine and feces. It's hard to imagine a disease, as Matt said earlier, that causes such complete loss of dignity, and the disease burden for patients and their families is huge. So it's estimated that approximately 50,000 hereditary and somewhere between 200 and 500,000 patients with non-hereditary or wild-type ATTR amyloidosis worldwide, although in truth, the prevalence is really not known. What is known is that the condition, particularly wild-type ATTR amyloidosis, is being increasingly diagnosed. The graph on the bottom left of this slide shows the number of new diagnoses of ATTR amyloidosis in the UK by year, and the trend that you see here is being seen all over the world. It's due to a combination of greater disease awareness amongst healthcare professionals and improved diagnostic imaging techniques, such that the diagnosis is less frequently missed than it was before. In addition, the diagnosis of ATTR amyloidosis is being made at an increasingly early stage of the disease natural history. The two figures on the right show UK patients diagnosed with cardiac ATTR amyloidosis over the last 20 years, divided into blocks according to year of diagnosis. Across time, you see an increasing proportion of patients diagnosed with early stage disease, which translates, despite the absence of disease-modifying therapy in all of these patients, into improved survival from the time of diagnosis. And this is also reflected by serial functional evaluation in patients diagnosed with cardiac ATTR amyloidosis, as you can see in the image in the middle there. In 2014, when the ATTR-CA study was recruiting, the decline in 6-minute walk test distance over 12 months among patients on placebo averaged more than 50 meters. By 2017, it averaged just under 50 meters, and in 2019, among patients with cardiac ATTR amyloidosis enrolled into the placebo arm of the ATTRibute-CM study, it averaged less than 10 meters. Some key facts learned from imaging of amyloid. More amyloid results in worse outcomes, less amyloid results in better outcomes. Natural clearance of in vivo amyloid deposits is always slow, although the rate of amyloid clearance does vary between individuals. The principle of treatment in all types of amyloidosis at present is to attempt to slow ongoing amyloid formation. This can be achieved by reducing the concentration of the circulating amyloid fibril protein precursor, in other words, reducing the substrate for new amyloid formation. The approved drugs which reduce concentration of circulating TTR protein are the so-called gene silencers inotersen, patisiran, and vutrisiran. Or specifically in ATTR amyloidosis, slowing ongoing amyloid formation can be achieved by stabilizing the TTR tetramer, again, thereby reducing the substrate for new amyloid formation. The approved drug which stabilizes TTR is tafamidis. There is hard evidence in all types of amyloid that have been adequately studied of a strong relationship between the magnitude of knockdown of the relevant amyloid fibril protein precursor and patient outcomes. The top right panel that you see there shows patient survival in AL amyloidosis stratified by the magnitude of knockdown of the amyloid-forming monoclonal light chain, with complete knockdown shown at the top there, patients doing best, and little or no knockdown doing worst at the bottom. The lower right panel shows the relative risk of death in patients with AA amyloidosis stratified by octiles of SAA concentration. Similar data are emerging in ATTR amyloidosis. In other words, the greater the TTR knockdown, the better the outcome, although these data are at present less mature. The TTR stabilizer tafamidis was first shown to slow progression of neuropathy in patients with hereditary ATTR amyloid polyneuropathy and was licensed in the EU, but not in the US in 2011 for treatment of hereditary ATTR amyloidosis with polyneuropathy. Tafamidis was subsequently tested in patients with predominant cardiac ATTR amyloidosis resulting in heart failure in the seminal ATTR-ACT trial. The primary endpoint was a hierarchical reduction in all-cause mortality and frequency of cardiovascular-related hospitalizations at 30 months. And this was met, and the trial showed a reduction in each of the individual components of the primary endpoint in patients receiving tafamidis compared to those on placebo, along with a slowing of the functional decline and slowing of the decline in quality of life as measured by 6-minute walk test and KCCQ, respectively. In 2019, tafamidis was licensed for treatment of patients with ATTR amyloid cardiomyopathy. The gene silencers patisiran, inotersen, and more recently vutrisiran were initially tested in patients with hereditary ATTR amyloid polyneuropathy in trials with a similar study design, although it's notable that the more recent vutrisiran trial used the placebo patients from the earlier patisiran trial as a comparator group for the primary endpoint. All of these drugs reduce circulating TTR concentration by an average of between 75 and 85%, and all 3 trials met their primary endpoint by showing a difference between the change in mNIS+7 neuropathy score from baseline to 18 months among patients on drug in whom the score stabilized or even improved, compared to patients on placebo in whom the score continued to increase, indicating worsening neuropathy in line with the disease natural history. These 3 drugs are all licensed for the treatment of hereditary ATTR amyloidosis with stage 1 or 2 polyneuropathy. Patisiran has subsequently been tested in a trial of patients with predominant ATTR amyloid cardiomyopathy and was shown to also benefit this patient group. So I think we'll discuss most of the challenges that are mentioned here during the course of the day. However, I believe that they're worth briefly highlighting in this introduction. So I believe that the trend of earlier diagnosis is set to continue. And this means larger and longer studies unless we develop better predictors of disease progression or disease improvement that can reliably be utilized in trials as surrogate endpoints at earlier time points. Secondly, the separation into polyneuropathy and cardiomyopathy trials, which is a product of the trial design requirements for licensing of drugs, has meant delays in licensing drugs that are ultimately treating a single multi-system disease. We have yet to find a drug that benefits the polyneuropathy and not the cardiomyopathy, or vice versa, which indicates to me anyway that there is probably unnecessary duplication and delay that could certainly be addressed. And finally, given the current costs of the relevant drugs, how are we ever going to compare drugs in head-to-head trials or test drug combinations? So I'd like to finish by thank you for listening. I hope to have given you some food for thought and look forward to a stimulating day. Thank you very much.
Oh, Julian, thanks so very much. That was perfect. We have two discussants today, along with Julian and myself, Michael Polodefkis from Johns Hopkins and Prem Soman from University of Pittsburgh. Maybe I'll start with a difficult question to Julian, not prompted. You know, people have regrets in life. Do you think of any scientific regrets that we should have potentially done things a little differently over the last decade related to transthyretin amyloidosis? Or maybe that's a shining light for what we could do better in the future.
So many things, so many things. I mean, yeah, it's a difficult question to answer, isn't it? Because there are lots of regrets. I'm sure we could all individually and collectively have done better. I think, you know, this probably having this meeting now in one sense is wonderful, but it's also a regret that, you know, we didn't come together as a group at an earlier stage and try and, you know, think carefully about trial design and licensing of drugs, you know, at a much earlier stage. So of course that is a, you know, that is a regret. In terms of the science itself, there are so many things, there are so many gaps still in our knowledge, aren't there, in terms of, you know, why do certain patients regress their amyloid more quickly than others? Why do patients get this disease in the first instance, you know, who don't have a mutation? What is the relationship with gender? You know, wild-type ATTR amyloidosis appears to be predominantly a male disease. There are so many questions that we really don't know the answer to yet. So, you know, good fodder for future research, but lots of regrets in terms of not knowing the answers already.
Yeah, I would echo many of those sentiments, maybe comment that, you know, we've, I think for clinical reasons and regulatory reasons, appropriately or inappropriately dichotomized the disease. And as you pointed out, many of the therapies work across the spectrum and the biology, whether you have a predominant, you know, neuropathic phenotype or cardiac phenotype seem to be effective. And I wonder if we could have, and going forward, if we could figure out a way to be a little bit more inclusive in our trial design to make sure that we are bringing along all the patients. Michael, maybe I'll ask you. There's been a great interest, obviously, in the cardiac realm. What are you seeing in the neuropathic realm? Have we solved that problem, or what are patients still presenting with and concerned about, and where do we go forward?
It's been transformative what's happened over the past, 5+ years. But as you alluded to, some patients continue to progress, and the leading question or desire is to improve, to undo damage that's done. And then close behind those questions are some of the things that Julian alluded to. How do we better predict the future for those who have the disease? How will they progress? For those who are carriers, who will get it, who won't, and how that will take place. And I think cost is a big concern. It seems with increasing frequency, we have denials and reapprovals, and the cycle of endless paperwork and anxiety. So those are certainly things that are at the forefront with the neurological aspects.
I think we've made tremendous headway, as you're saying, and the anxiety of securing therapies is always a concern for all the patients, and we'll spend some time, I think, this afternoon talking about that. I do want to encourage anyone from the audience there to get up if you have a question or want to engage, anyone online. Prem, we heard from Julian that, the disease is, from the cardiac perspective, being diagnosed much earlier, and I think that's a testament to, as Julian elegantly showed with Mariana, non-invasive imaging. Maybe you want to comment a little bit further. Have we, solved all the non-invasive imaging? Where are the gaps a little bit in that regard, for particularly the design of a future trial?
So thanks, Matt, and good morning, everybody. So my name is Prem Soman. I'm from the University of Pittsburgh, where I run our nuclear cardiology program and also the Amyloid Center. So I come at this from as somebody who does imaging every day, but also treats these patients. And I will start by, at the risk of repeating what Julian said in his elegant talk and stating something that's completely obvious to everybody, state that, this field is so unique in that there have been sort of breathtaking developments in a short time, and yet there are so many fundamental gaps in our knowledge. And one that I will bring up to answer Matt's question, and that's probably relevant to what we're trying to do here today. I think as I treat these patients, one glaring gap in knowledge is we don't quite know what happens in the myocardium when we treat amyloid. What underlies the clinical improvement? Is it just that they don't deposit more amyloid? Is it some sort of resorption of amyloid that happens? Or is it something completely different, like a toxic component, such as the like that we know exists in AL amyloid? And I bring this up because if our objective is to come up with metrics of disease regression or progression, it's really important to understand what happens in the myocardium so that we can identify imaging metrics or clinical metrics that parallel the pathophysiology of what happens with therapy. And I think the signals from imaging have been quite conflicting. Now that we have anecdotal reports, for example, of improvement in PYP scintigraphy, what is very obvious is that the parallel changes, for example, in MRI and echo are not the norm, right? You see the scintigraphic intensity reducing, but it's rare, I think, at least in, and I'm asking this as a question, to see regression of LVH on echo. And in fact, the correlation between what an echo looks like and what a patient's doing clinically in amyloid is not that clear. So I would bring that up as one important gap we need to address to be able to define metrics of response in these patients. And again, this plays into, when is it futile to initiate therapy? Is there a point at which it is too early to initiate all these expensive therapy? And can we get some insights from imaging on these important questions? Thank you.
Yeah, maybe Julian, you could comment, but we've talked for a long time about amyloid versus amyloidosis. Are we going to end up finding individuals with such early disease that may not progress over time, that maybe watchful waiting is appropriate? Was that a potential possibility?
I think it's an absolutely excellent question, isn't it? And it's an unknown, really. As many people in the room will be aware, post-mortem studies have shown that up to 25% of individuals over the age of 80, have ATTR amyloid deposits in their hearts. And what we really don't know is what proportion of those individuals actually had a significant amyloid-related cardiomyopathy and what proportion just have incidental amyloid deposits in their hearts that don't cause clinical disease. And obviously, one end of the spectrum would be many, many millions of patients with, amyloid cardiomyopathy, and the other might mean far fewer. So I completely agree, Matt. I think we really don't know where we are on that spectrum. I think what we, and we have a very poor handle at the moment on the natural history of very early stage disease. And I think, we know that when a patient presents with significant cardiomyopathy, they invariably progress. And, I think we're all, as clinicians, aware of that. In the absence of treatment, they invariably progress. But what we really don't know is, if one detects, if one is able to detect very, very early stage disease with a few ATTR amyloid deposits in the heart, will those patients necessarily progress? And if so, at what speed? So I completely agree. I think those are questions that we don't at the moment have the answer to. And, as again, many people are aware in this room and are involved with, there are many studies trying to look at exactly this question of the natural history at the early stages of disease.
Yeah, I'm also curious for people's feedback, where we've gone from a disease where there was no treatments to now one where we're, I think, in an enviable position of talking to patients about different therapeutic options. I'm curious how people, either on the panel or talk to patients a little bit about this and making choices and choosing among the different options. We touched a little bit on combination therapy. Is that something that you're seeing in your practices? So maybe go around, Michael, or you. How do you have this discussion with patients about whether to pursue a silencer or a stabilizer?
Well, from a neuropathy standpoint, we're limited to a handful of therapies, but the combination of silencers and stabilizers is a difficult one. And usually what happens is the cardiologist prescribes a stabilizer and the neurologist may prescribe the silencer. But when to initiate dual therapy, I think remains an enigma. And I think the general strategy is one is started, and if there's evidence of clear progression, the second is started, but that's done a bit blindly.
Maybe on a similar vein, how do people have this conversation about which trials to participate in? We used to not only have one trial at a time, but now we could have several. Anybody? Okay.
So, I think in terms of therapy for predominant cardiomyopathy, in my practice, we choose tafamidis as the primary therapy. I can only think of one patient who's on combination therapy, and it was a young woman who years ago had a liver transplant, had neuropathy, and then had progressive cardiomyopathy wild type on a silencer. So I added a stabilizer. I can't recall any other patient that we've had dual therapy on.
And just returning to the previous conversation, I think, Sharmila, you had a comment you wanted to make about, I think, imaging.
Yeah, thank you, Matt. So 2 comments. One is what Prem Soman mentioned is so true. That is, we are seeing changes in PYP maybe without changes in echo and MR. This is not new to the field of oncology, for example, where FDG signal may improve before you see overt structural changes on CT or MRI in the tumor. So the way I would like to look at it, of course, none of us know this, is that the change in PYP signal may be representing a molecular change in amyloid that we are yet to understand, which may be one step ahead of real improvements in MRI or echo. So I think we need to be open to that possibility. The second thing is Julian's point about, and your point, Matt, about amyloid versus amyloidosis. I think detection of early amyloid deposits may be very important because one of the questions that we need to know is why are some patients with amyloid deposits resilient to develop amyloidosis? And that may be a key to some of the therapeutic advances. Like if we can find out what's protecting them from progressing into overt infiltrative process, as opposed to others who have deposits and they go on, that may be important information and needs for this study.
Yeah, I would agree. I mean, Julian, maybe you want to comment, but your recent seminal publication with Mariana about patients who spontaneously reversed certainly suggested some therapeutic strategies. So natural history studies that potentially observe resilient patients who aren't progressing could provide us with some insights. Any thoughts?
Absolutely. Thank you, Matt. Yeah, we recently discovered 3 patients who very remarkably spontaneously recovered back to pretty much completely normal health. These were 3 patients who presented very typically with cardiac ATTR amyloidosis, and a year later, were reporting very marked improvement. And when we looked at their imaging, quite remarkably, particularly their cardiac MRI scans, we saw a very dramatic reduction in the MRI abnormalities, but which were also accompanied by a very marked reduction in wall thickness, which I think is worth mentioning. There were 4, 5, 6 millimeter reduction in wall thickness on echocardiography, and marked improvements in biomarkers, etc. And obviously, with these 3, very unusual patients, we tried to determine what might be causing their clinical improvement, and we found that all three patients had antibodies directed or had developed antibodies directed against their ATTR amyloid deposits. And when we looked at 350 patients with cardiac ATTR amyloidosis who had progressed in the usual way, we didn't find any evidence of antibodies in those patients. Similarly, one of the patients actually had an endomyocardial biopsy, and in that patient, we saw an inflammatory response, which was reminiscent of the inflammatory response with macrophage infiltration that we saw in a mouse model that we treated with AA amyloidosis some years ago with antibody therapy, and in which we were able to achieve rapid reduction of amyloid in this mouse model, and that was subsequently achieved also in humans, in human visceral amyloid using an antibody. So we believe that if one can achieve if one can get antibodies into amyloid and antibodies that activate complement and cause an influx of macrophages, that may well lead to rapid resolution or rapid regression or removal of amyloid, which is an exciting prospect. It's obviously a very rare natural phenomenon that we've recently discovered, but certainly gives cause for optimism in terms of future treatment.
So I thank the discussants and Julian for an excellent presentation that started us off, and I'm happy to turn the mic over to Dr. Prem Soman, who will be talking and leading a session on changes in the phenotype, shift to earlier stages, and how that's going to affect our future clinical trials.
Thank you, Matt. It's my pleasure to introduce Maz and Hannah. We all know Maz. Maz is a co-director of the Amyloid Center at the Cleveland Clinic, and then will lead the discussion on the second session of the day, which is titled, How Will Shifts in the Stage of Patients with ATTR-CA Affect Ongoing and Future Clinical Trials? Maz.
Thank you, Prem, and it's really an honor to be here with all of you today. And my talk, which is How Will Shifts in the Stage of Patients Affect Ongoing and Future Clinical Trials, will have a lot of overlap with Dr. Gilmore's talk. These are my disclosures. But if we look at the timeline of where we were, are, and going with respect to phase 3 clinical trials in ATTR cardiac amyloidosis, it starts back with the TRACK trial. And we enrolled patients between 2013 and 2015. And it wasn't until about 6 years later, right around the time of FDA approval of tafamidis, that we started enrolling the ATTRibute cardiomyopathy trial, testing acoramidis. And then a year later, in 2020, the 3 main gene silencing trials, Apollo B, Helios B, and CardioTransform, started to enroll. And now we have some 12-month data from Attribute and Apollo. We're still awaiting more definitive data for a lot of these trials, and we're going to be embarking on several other phase 3 randomized trials within the next year. And what's evident over the last 10 years is that patients we're diagnosing and enrolling are different from 2013 till 2023. Insofar as the patients that we're enrolling in ATTR-CA were more at a later stage of disease with respect to functional class, biomarkers, and now we're enrolling people, so to speak, in the intermediate stages, relatively speaking. And this is where I'm just going to highlight the National Amyloidosis Staging System, NAC Stage 1, 2, and 3. So with their large cohort of ATTR cardiac amyloidosis patients, the NAC derived a staging system with two simple parameters, NT-proBNP and an estimated GFR. And stage 1 is when your NT-proBNP is less than or equal to 3,000 and a GFR greater than or equal to 45. And stage 3 is greater than 3,000 and GFR less than 45. And if you look at the median survival, 69 months for stage 1 and 24 months for stage 3. And so Dr. Gilmore just showed you this, their 20-year experience with nearly 2,000 patients with ATTR cardiac amyloidosis broken down into 5-year intervals. And again, if you look at the last interval, which is 2017 to 2021, 56% of patients were diagnosed with NAC stage 1, whereas previous to that, it was in the low to mid-40s. Also, if you look at stage 3 patients, so more advanced patients, NAC stage 3 was 18% previously and down to 11% in the last cohort. Looking at Columbia Amyloidosis Center data, and this is courtesy of Matt Maurer, they looked at 419 patients with ATTR and also put them into different areas and used their Columbia staging system. And as you can see, over the different eras, the percentage of patients diagnosed with Columbia stage 1 was nearly 50% in the last cohort. And again, for Columbia stage 3, going from 27% down to 8%. So we're clearly diagnosing these patients at an earlier stage of disease. Again, Julian showed this, but 2017 to 2021, which is the last cohort, at 30 months, there's an 80% survival, and this included censoring patients who are in clinical trials or getting disease-modifying therapy. You'll hear more about this in the afternoon from Pablo Garcia-Pavia. But if you juxtapose that with the survival in the ATTR-ACT trial, at 30 months, the placebo survival was 57%, and even in the treated patients, it was 70%, so lower than we saw in the NAC natural history most recent era. And you can see on the left the patients that we're enrolling, NT-pro about a 3,000, EF 48%, et cetera. And when you look at the enrolled patients, the baseline characteristic in ATTR-ACT versus the last era of the NAC natural history, you can see that the NYHA3 patients, 30% versus 11%. The 6-minute walk at baseline was higher, the NT-pro lower, despite similar echocardiographic variables. Now, with respect to disease trajectory in ATTR-ACT, we all know tafamidis abated the decline in 6-minute walk in KCCQ. And if you look at 12 months, in the placebo arm, 56-meter decline. And even in the tafamidis-treated arm, there was a 23-meter decline at 12 months. And with respect to the KCCQ, there was about a decrement of about 11 points in the overall summary score at 12 months. Now, as far as the Attribute cardiomyopathy trial, their primary endpoint at 12 months was the change in 6-minute walk. And as we know, they did not meet the primary endpoint at 12 months. You can see there the reduction in 6-minute walk, acoramidis versus placebo. But what's important to note is that if you look at the Attribute cardiomyopathy placebo arm, they declined at the same pace as what you would expect from a healthy elderly adult over 12 months. Clearly, patients with a different trajectory than we saw in ATTR-ACT. And again, here you can see juxtaposed the NAC natural history decline of 45 meters, the ATTR-ACT placebo 56 meters in 12 months, and again, the treated ATTR-ACT patients 23 meters. And finally, if you look at the Apollo B trial, they randomized 360 patients to patisiran or placebo. A quarter of patients were on tafamidis. And again, primary endpoint at 12 months was change in 6-minute walk. And here you can see the decline at 12 months in the placebo arm was about 21 meters. again, the decline in the KCCQ was about 3.5 points at 12 months. So if we look at these characteristics of the ATTR cardiac amyloidosis trials to date, first looking at the baseline characteristics, you can see on average the patients are a little older, but the proportion of patients with NYHA3 has progressively reduced. The baseline 6-minute walk is higher, the baseline KCCQ is higher, and the NT-proBNP is significantly lower. And as far as disease trajectory, the change in placebo at 12 months, as we saw, the placebo ATTR patients, 56 meters in 1 year versus 7 meters in 21, respectively, for the latter two trials. And again, the change in quality of life, KCCQ score, 11 points at 1 year versus 3.5 or so at 1 year. So as Dr. Gilmore stated, and this was really a lot of reiteration, we are diagnosing these patients earlier, and the patients we are enrolling in these trials are at an earlier stage of disease. As such, harder endpoints like mortality, CV hospitalization, et cetera, may be more difficult to meet because, again, of less severe baseline characteristics, a slower disease trajectory, the fact that we now have background disease-modifying therapy that will further alter that trajectory, and thus the time needed to show difference will be substantially longer. And I'll just finish with improvements in quality of life and functional capacity are extremely important for our patients. Thank you.
Thanks, Mass. And thank you to you and Julian before you for setting up the stage for what we are charged with discussing in this session, which is whether these shifts in the stage of patients, i.e. detecting disease earlier, how they're going to affect future clinical trials. Before I do that, let me ask Michael, are you seeing the same sort of early stage detection in the neuropathy world?
Yeah, absolutely. And that's echoed in the different trials. The patients in the original Apollo trial tended to be more severe than the subsequent Helios trial. But they're the same themes, yes.
So perhaps as a jumping off point for discussion, I'll pose this question, which is, can we do placebo-controlled trials anymore, either as randomized trials or even with a historical placebo data as the comparator? I think, Martha, you're online and raised your hand, so I will pick on you if I may first.
Oh, okay. Yeah, I had actually raised my hand a little bit earlier. So as far as placebo-controlled trials, I don't see how we could justify those. I don't think there'll be a role. The earlier stage is a big challenge, and as Julian mentioned, it's not necessarily what people want to hear, but because this is such a generally slowly progressive disease, especially with earlier stage patients, unless we come up with really fantastic surrogate endpoints, we will need longer trials, not shorter trials. So when we start looking at 12 months, it's not a big surprise that we're not seeing a statistically significant change. And I know that's not what sponsors want to hear, longer trials, but clinically, we're really going to need longer trials, I think, to know which treatment options are most efficacious. So I think that's a big challenge. One thing I want to jump in here, no doubt that because of raised awareness, we are capturing patients earlier. However, patients that we label as early by biomarker stage, the NAC staging or Mayo staging, doesn't matter. Many of those patients, a lot of them actually have very advanced structural heart disease. So this is not just like a little bit of cardiac dysfunction. Many of them have, that's that disconnect, but they are still pretty well compensated. So then even earlier disease, that's probably more challenging. The last comment that I have is that although probably the majority of our patients diagnosed at earlier stage are due to improved techniques, non-invasive diagnosis, and raised awareness, we have this very significant problem, at least in the United States, of patients being misdiagnosed with amyloidosis when they do not have amyloid. So that's a really big problem with misinterpretation of PYP scans.
Thank you, Martha. So, we have representatives from the industry, from the regulatory field. So how does this affect sort of drug development from an industry perspective, having to do these really large trials and long trials with hard endpoints? Anybody from the industry like to comment on that? Or from the regulatory organizations?
Prem, can I jump in here?
Yes, please.
So I'm neither industry nor regulation, but I come at it as a real-world evidence methodologist. I think that perhaps we have to challenge ourselves into saying we've gotten to a point where we can no longer think of our amyloid patients as a homogeneous group, and can we rely on statistical developments in heterogeneous treatment effects to start and identify, even within the early stage people, individuals who are likely to have either at a greater risk of events or a greater risk of progression or a greater benefit from the therapy. The reason why this is harder than it sounds is so far we've lived in this world where we run these randomized trials of drug A versus placebo or drug A versus drug B, and often these data sit with sponsors. And we'll have to figure out a way to pool these data across these different trials or even the observational data to be able to do the HTE methods. I'll just leave you with the idea that the HT heterogeneous treatment effect methods have changed substantially in the last 2 years, and that there may be an opportunity for us to be able to use these in amyloidosis.
Thank you. Yes, please, Adrian.
Yeah, I guess 2 comments. One, just on the placebo concept. So one way that certainly you could see life science companies attacking that problem is go to places where tafamidis is not available. And so I think that's something, for the day that, we need to really be considering, hey, do we want to ensure that we have evidence generation that certainly is representative of all communities, but also including the communities that have access to tafamidis, and so what are designs for studies that would address that combination of issues, those that don't have access versus those that do, in terms of different countries. The second thing is that, I know, probably later in the day we'll get to discussions around different trial designs, but if, places have tafamidis as their baseline or standard therapy, you could also imagine trial designs that include tafamidis withdrawal. And so, eventually seeing, if you can develop enough data in terms of placebo control, also thinking about designs that include tafamidis withdrawal if there's a new therapy being considered.
Thank you.
It looks like Pablo wanted to make a comment. I'm just noting.
Yeah. Hello.
Pablo.
Yeah, hello. Thank you very much. Well, thank you first of all for the invitation. And regarding all what has been said, I wanted to point out that although I agree, and it's completely clear that we are diagnosing as a whole the patients earlier, the number of patients we are diagnosing, as it has been shown, is increasing exponentially. And the number of patients who are sick or who have an advanced phenotype is increasing, in absolute numbers. So we should not forget about that, in the sense that this is an increasing problem. And I think with aging will still increase. And the other comment that I wanted to make was briefly said before as a question regarding if we should, as a community, try to stop the placebo-controlled trials. Because at this stage, what is happening, in my opinion, is that we are going the pathway that we are maybe following the pathway that we had in other cardiovascular areas, like the heart failure trials, that we have kept adding different agents to an standard therapy, and maybe we are in a situation that we should compare one to another instead of adding more and more, which is going to complicate treatment with patients because we're going to end up with a number of different therapies, all very expensive, and maybe the additive effect of some of these are going to be very little, and that's something that I thought it was worth bringing to a stage.
So Pablo, you just reported on a phase 1 trial for the antifibrillary therapy. So how are these observations sort of changing how you're thinking about, phase 2, phase 3 trials for that particular agent?
Well, what I think is that it will be very important to think what's the next steps, and obviously I think that in that sense the regulators play a pivotal role, because the industry always have to work very closely to them to see what to do and what to propose afterwards. But certainly I'm trying to look what's going to happen from the day, in 3-4 years time, and with this changing landscape of therapies, of the patient population that we are facing, it's going to be very difficult for clinicians as me to decide which therapy is more appropriate for our patients, because we're gonna end up with different trials, with different drugs that have not been compared head to head. So it's certainly something that regulators should think about, because otherwise we're going to follow the path of adding, and again, showing a benefit in a trial compared with an existing therapy obviously is risky for the industry, but at the same time poses an opportunity to undertake easier trials with better possibility of showing differences, because you're not going to have a baseline treated individuals.
Thank you. Martha, then Emmanuel, and then Maz, you have a comment. So in that order, please. Thanks.
Great, sure. And I'm Martha Grogan, a cardiologist from Mayo Clinic, Rochester. They've asked us in the chat to identify ourselves. I might let the organizers know there's quite a few questions and things in the chat. Just to speak to Pablo's question about, therapies and design of trials, especially combination therapies or choice of therapies. This is, of course, a huge dilemma, probably even more so when we have an earlier stage patient. But within the current structure of clinical trials and industry-sponsored trials, I don't see any real easy solution to that one, because where is the motivation going to be for one sponsor? There's not going to be a lot of interest in doing a head-to-head comparison, but it leaves us with clinicians with really more questions than answers. I do wonder, because I really suspect that unless there's just a paradigm shift, we are going to learn this more from experience as we get more drugs on the market. I think it's really honestly going to be our clinical practice. And I know that we don't usually have this so much for drugs, but for devices, sometimes there are mandatory or near universal registries where we can really ensure that we study this in the real world. And I'm afraid that might be how we get some of these answers. But right now, combination therapy is a huge question mark, and I don't think we're going to have the science anytime soon. The other concern about, I can understand the proposal of considering a placebo-controlled trial in areas where there isn't access to medications, but as Pablo's alluded to, I think as a community, we really want to remove that barrier. Ethically, I hope that we can really try to get access to these transformative therapies worldwide, rather than use a segment of individuals for placebo-controlled trials.
Thanks, Martha. Emmanuel?
Thank you. I'm Emmanuel Zirdex. I'm a medical assessor at MHRA London. And thank you for the invitation. And thank you also the presenters for the very interesting data and new evidence. What I would like to point out is that what came out from the previous slides is that obviously with our knowledge about the disease is changing, and we have the data from tafamidis and the licensing of tafamidis, which, as everybody says, has changed the landscape. However, taking all this together, it becomes very evident that although tafamidis had somehow broad inclusion criteria, the population that was studied in ATTR-CA may not be representative any longer of the diseases we know today. So one question, of course, is if if the ATTR-CA trial started today, would it have the same outcome? And that will all of us will need to keep that in mind, that perhaps when we think about future trials and comparisons with the ATTR-CA or how to deal with the licensing of tafamidis now, to make sure that we compare similar populations and similar stages of the disease. Tafamidis has a broad license indication now, but that was based mostly on extrapolations because the populations, as we saw in the slides, was very specific and again may not represent the diseases we know today. So I think this will need to keep in mind both for clinicians and regulators.
Thank you, Emmanuel. I know Maz is waiting, but Emmanuel asked a very provocative question, and maybe I'll direct it to Matt. If we repeat ATTR-ACT today, do you think you would have got the same result?
I don't know. And what we do know is that tafamidis is a more effective therapy earlier in the course of the disease. Then again, if your placebo group doesn't have any events, who knows? So, the relative risk reduction in mortality for the very few subjects, there were 37 out of the 441 in the ATTR-ACT trial who had New York Heart Class 1 symptoms, was 64% reduction in mortality. So the relative risk reduction was even greater than in Class 2 or 3 patients. So it could be that there's a convergence of good news, right? Earlier patients, these therapies, my experiences work better in patients who have less organ dysfunction. So I don't think we should beat ourselves up too much. This field has, moved in a very good way. We have some new challenges, but I think it's great that we're diagnosing people earlier. But whether it would work, I don't know if it would meet all the metrics. But I would comment that, a little luck helps too. Thanks to the FDA, they mandated that we had 30% variant patients in ATTR-ACT. We didn't. We had 25%. We worked really hard to get 25%. When we hit 400, we didn't have 25%. We closed enrollment to wild type and only recruited variant patients. As we know, variant patients progress much more rapidly than a wild type patient. So thank you to FDA for making that suggestion. They also suggested, in meetings, why not use a higher dose, 20 versus 80 milligrams. And, we designed the trial originally. We thought 24 months would have been enough. Thank God we went to 30 because we wouldn't have met the metric either. So, in hindsight, you sometimes make a very good or intelligent, or in Mike, I think this case, lucky decisions about how you design these particular trials.
Matt, and then Clemens.
Yeah, I'll be brief. I just want to echo Pablo's comment. So if you're at any larger volume amyloidosis center, you're still going to see, more advanced patients, NAC stage 2, 3. Unfortunately, they're there. And, there's this tension with all these clinical trials of wanting to only run it a certain period of time and get the drug out there and get the results versus doing a longer trial where we're much more likely to see benefit. I think that that's the tension that we're all going to talk about. The trials that are coming are going to have NT-pro BNP inclusion criteria that are going to be higher, 1,000, maybe 2,000, 3,000. And so there's going to be patients that are not going to be able to get into those trials because they're trying to enrich the population for endpoints. And, I would kind of end with just stating that, this is the elephant in the room, but there's ongoing trials overlapping. And, we're going to have some patients that are in a trial. They may be in an open label extension or still finishing the trial. And then another trial comes up and they're like, well, can I go into that trial? So that's a real, actual potential issue. I hope it doesn't occur, but I just kind of wanted to throw it out there.
Thanks, Matt. Clement?
Just two short notes. One is, I'm Tom Smitman. I'm from the Federal Institute of Drugs, Medical Devices in Germany, and I'm also involved in EMA activities, but I'm representing personal views and not the EMA view. Regarding the placebo-controlled trials, of course, placebo-controlled trials are difficult or impossible to conduct if patients on placebo don't get treatment. However, if you're dealing with a new mechanism of action like depleters, so then you can conduct placebo-controlled trials as long as the patients can get everything that is approved, like silencers or stabilizers. So in this area, placebo-controlled trials, I think, are feasible. And to make them feasible, you have to, even more feasible, you have to plan your study very well. And in this regard, the knowledge of the natural history and your staging systems are kind of helpful. The better you can characterize your patient population by these staging systems, the lower will be the variability, and the higher will be the probability that you have a nice outcome. So moving away from neuropathic association stage and towards these more refined staging system is something that is important. Regarding use of historical placebo controls, as such, I'm a little bit skeptical from the regulatory view. The usual way you do that, incorporate that, is the non-inferiority trial that may address what has been mentioned by Dr. Pavia. We as regulators are not opposing head-to-head comparisons or non-inferiority trials. In these non-inferiority trials, we incorporate knowledge about placebo arms when defining the non-inferiority margin. We have to take into account that the placebo arm is the assumed effect compared to placebo is a moving target. Placebo effect today is not the same as at the time where the active comparator were used. But it's not the regulators that are opposing non-inferiority trials. It is more maybe it is more the industry that for some reason, for a reason we can understand, don't want to go that way, but it's not the regulators. Thank you.
Thanks, Clement. Sam, you have your hand up.
Yeah, hi everybody. I'm Sam Tsimikas. I'm a cardiologist and I represent Ionis. I just wanted to make a brief comment on the head-to-head comparison, which is a great idea, but at the practical level, it's very difficult, particularly for small companies. And just to give you an insight, we considered doing this for the eplontersen trial that's ongoing right now, CardioTransform, but we have to pay for tafamidis, and at over $200,000 a year for 3 years, it ends up being about $700,000 per patient. And so it's really prohibitive to be able to do that kind of head-to-head comparison. So you have to use the alternative, which is to go with on top of standard of care. So that's what drives a lot of the decisions, particularly for small companies. Outside of the risk, the cost is really, really a lot for doing that kind of trial.
Julian, you have your hand up, but before you ask your question, can I also ask you to comment on, so we've been talking about clinical trials, but, following natural history of patients, as you mentioned, is also become difficult because of early diagnosis and the evolving therapeutic landscape. So, you know, we start treating patients early on, and they appear to sort of live longer, but how do you differentiate sort of a lead time bias effect from a true effect on mortality?
I couldn't agree more. I mean, I think you know, that's a real challenge, and lead time bias is going to be a huge problem if we're studying these you know, these early stage patients. So I couldn't agree more. Challenge. I don't have an answer. I wish I did. If I can perhaps just make an, you know, draw on experience of oncology. You know, there are very few cancers, aren't there, that are, you know, that are treated with a single drug. And conceptually, at least, it's very hard to imagine that the combination of a drug that slows amyloid production, for example, a stabilizer or a silencer, you know, in combination with a drug that potentially accelerates removal, like NI-006 that Pablo recently published on, it's very hard to imagine that, you know, using those two drugs together won't bring about added benefit over and above one or the other. It's not to say that, you know, using one or the other, you know, may overcome the effects of the other, but certainly conceptually it's attractive to use both. So I think it is important that we, you know, devise or think about means of potentially testing drugs together, combinations of drugs in trials, as we've talked about.
Thanks, Julian. Preston, you had a comment.
Actually, thank you so much. It's more of a question for Matt and for Sharmila. In this day and age of advanced analytics that I live in now, there's much interest in looking at characteristics of patients that might predict rapid progression for making clinical trials more efficient, specifically multi-omics, imaging, demographics. Has anyone discussed, or approached the idea of, say, taking the ATTR data set and getting somebody really good at machine learning to go analyze this, to try to look at your class 1 and class 2 patients to try to identify who's going to be a rapid progressor, so you might improve your event rate in a trial to treat some of these more rare conditions?
Yeah, I think it's a great suggestion. Unfortunately, it hasn't been done. And just going back to Dr. Kazi's comment from a while ago, if I had a regret, it's that we don't leverage these data sets that we have. And I would encourage everyone who's in an amyloid center, also those in industry, to figure out ways where these data sets can be leveraged by very smart people with, as you're saying, new scientific approaches in the omics realm or machine learning to better identify relevant cohorts that could facilitate, drug development. Sharmila.
Yeah, I could not agree more. I think it's a great comment, Preston, and a very important comment in that it's not only applicable to identify rapid progressors, but in my mind, it's also important to understand non-responders to therapy. Within the tafamidis trial, we know that 30% of the patients on treatment did not survive at the end of 30 months. So basically, how do we do this how do we minimize futile therapy? How do we give them an alternate drug which may potentially save their life as opposed to continuing an ineffective drug? So it's a completely unexplored area that needs further work.
Go ahead.
Can I make a quick comment on this? I wanted to go back to Sam and Martha's comments from earlier. We've talked about learning from oncology. There's also an opportunity to learn from the cardiovascular devices trials. And I'll point out two things. One, to Sam's comment about how do we pay for the placebo arm, like it's the placebo arm or the control arm, if it's on tafamidis, so that's $250,000 a year. How do we pay for it? And there is precedent in the device world where if the control arm is getting a therapy that is considered standard of care, then one can work with Medicare to get Medicare to pay for that therapy without the intervention arm being standard of care plus a new therapy. So saying we're comparing A versus B, we have enough data for B, but will Medicare pick up the tab for A? So just, again, we could figure that approach from the device world. And to Martha's comment, it is in fact true that we haven't historically had registries for drugs, which are now very commonplace for CMS to say, you can't get device A unless you're also in a registry. This has, I think, at least partly been rooted in the fact that devices have been thought of as very expensive and drugs as something that are too inexpensive to justify a registry. I don't need to tell this audience that that is no longer true. And so I think pushing for universal use registries will also allow us to do heterogeneous treatment effects, understand disease progression that will allow us to enrich future trials, as you were pointing out, and then study who benefits from combinations versus not from individual drugs.
Thank you. I have my eye on the clock here. I see Cynthia Welsh from the rare disease space in the FDA, and I was wondering, would you like to comment on the issue with capturing, natural history of a disease with a changing landscape? And then Rick.
Hi, good morning. Cindy Welsh from the Rare Diseases team at the FDA. So I can comment and say that we want to be your partner in this. We want you to come in early and often and speak to the review divisions. We have a number of ways that we can help interact with sponsors and patient communities through complex innovative design program, pilot program, model-informed drug development program, rare disease endpoint advancement program, which applications will be available to be submitted for the RDO program starting July 1st through September 30th. And you can also use the natural history studies information to help identify different subgroups that you might want to study, inform eligibility, and look at safety analyses. And then the Rare Disease Cures Accelerator has a program for data analytics. And then there's also a new program that's starting with Clinical Outcome Assessment Consortium. I checked their website yesterday and that hasn't been posted yet, but they do have other resources on there. And, involve the patient community early to help find out what's clinically meaningful for them. And you can help use that to design your trial and to look at the duration of the trial to make sure that you're getting the information you need based on the, endpoint that you've selected. And if anyone has trouble, doesn't know who to contact at the FDA, you're always welcome to contact me or the Rare Diseases team, and we'll be happy to get you in contact with the proper review division. Thank you.
Thanks. And we'll be talking more about this in a session that's specifically directed towards endpoint choice later this afternoon. Rick, I think you get the last word.
That's wonderful. Thank you, Prem. Rick Ruberg from Boston. I will be quick because we have 58 seconds left. I just wanted to make a quick point about concomitant drugs. And our interpretation of the efficacy of our therapies, specifically ATTR, is going to be shaped by the medications that we use to treat the non-ATTR heart failure decongestion sort of medications. And taking a page from our other therapies, optimal medical therapy, guideline-directed medical therapy for other diseases, I think we need to pay a bit more attention to that, especially as we design these trials, and maybe stipulate specific concomitant therapies so that we don't wash out treatment effects or potentially bring our placebo patients, to a place where we are unable to see the effects of our drugs. Thank you.
Thanks, Rick. A really important point. Any other last-minute thoughts, comments? We've got a few seconds left. If not, thank you, Mass, for setting us up for a discussion, and thanks to everybody participated in this extremely informative discussion. Back to you, Matt.
Thanks. And our next discussions are going to be headed by Rosalind Adigan and Preston Dunman, and we're going to talk about what we can do collectively for non-cardiac ATTR drug development.
Thank you very much. Good morning. Once again, I'm Rosalind Adigan from the Department of Cardiology and Nephrology. So for this next session, we have one presentation and two perspectives where we will be hearing from some of our regulatory agencies from Europe and Japan, as well as some subject matter experts. So I would like to first start by introducing Dr. Jeff Kelly, who's going to give an overview of non-cardiac manifestations of TTR.
Okay, great. Good morning, everyone. It's a privilege to be here. I have a very short deck just to catalyze discussion. Is the slide deck up? Because I can't see it.
Yes.
Yep, it's up. We can see it.
Thank you so much. So I thought so maybe the most important thing I can say has already been said to a certain extent. That is, the TTR amyloid diseases are certainly multi-organ diseases, and we've heretofore done clinical trials largely focusing on a given organ system. And what I'd like to do today is to just remind everybody that the central nervous system and the eyes are also dramatically affected in these patients and often compromise quality of life. Next slide, please. So certainly there are rare mutations that present initially as central nervous system pathologies. Many of the TTR sequences that aggregate and lead to various organ-centric pathologies also cause eye pathology. And we're all very much aware of the cardiomyopathy and polyneuropathy. Next slide. So what we've learned, interestingly, from liver transplant, which of course was a therapy whereby the mutant protein was replaced by the wild type protein largely as a strategy to treat polyneuropathy, is that after about a decade, these patients who generally do better present with central nervous system pathology. So they present with dementia and focal neurologic episodes, which is loss of consciousness from a short period of time to fairly long periods. And it's now, next slide, please, quite well established that while liver transplantation is quite effective at slowing down polyneuropathy, next slide, it's that's shown here, next slide. It's clear that we are not affecting, of course, the source of TTR in the brain. So, of course, transthyretin is secreted into the blood by the liver, and it's secreted into the CSF by the choroid plexus. And as the tetramer comes out of the choroid plexus into the CSF, it can dissociate, misfold, and aggregate, leading to destruction of the central nervous system. And most likely the CNS pathology is slow to develop simply because the protein is 25 times lower in concentration in the brain. And remember, these aggregation phenomena are concentration dependent. So that's the most likely explanation of why the CNS pathology is slow to develop, whereas other organ degeneration phenotypes generally present earlier. My sense is that the central nervous system pathology probably exists in all TTR patients. We know much less about, what happens in a wild type TTR cardiomyopathy patient. As they live longer now, particularly on the silencers, we're going to learn about whether there are central nervous system pathologies. Next slide. So Yoshi Sikajima in polyneuropathy patients treated by liver transplant showed in the left side here by PET amyloid imaging that after about a decade, there's a lot of transthyretin amyloid in the brain. And on the right side shows that this correlates with an increased incidence of focal neurologic episodes. And in the following slide, Teresa Callejo and her colleagues showed that these individuals suffer from dementia as assessed by metrics that are quite commonly used in Alzheimer's disease. So if we could go to the following slide. We certainly didn't design tafamidis to get into the brain, and it doesn't do so very effectively, but there is about a stoichiometry of 0.5 tafamidis molecules to TTR. So I think over time, we need to learn whether tafamidis is going to be sufficiently effective in the brain and whether it's possible that other treatments could also affect the central nervous system pathologies. Where we have, in my mind, a complete unmet medical need is in the eye diseases. There are almost no therapies that I'm optimistic about getting into the eye. And so I think we need to start to think about that as we go forward. Certainly that dramatically compromises quality of life. I think I'm done. I hope I catalyzed some discussion, especially around the role of the central nervous system pathology in patients presenting with cardiomyopathy or lack thereof.
Thank you very much, Dr. Kelly. Next, we're going to have Dr. John Burke. Dr. Burke is a professor of medicine from Boston University School of Medicine, and I believe he's joining virtually as well.
All right, this is me. Following Jeff Kelly is an auspicious opportunity, but people generally don't keep up with his standard. There are a couple of comments I'd like to make. The first is that I'm a proponent of dichotomizing disease, and the reason is pretty straightforward. The original drug trials were intent on proving drug effect, and that's most rapidly done in a segment of disease that is rapidly progressive in the untreated population. Neuropathy progresses much more rapidly than cardiomyopathy and affords shorter trials. In fact, what we know at this point is neurologic impairment score without all the bells and whistles demonstrates drug effect at least by 12 months and probably by 9 months. And cardiomyopathy, we don't have measures of disease that demonstrate the progressive changes. And so we're at the moment dependent on long-term outcomes that are hard and irrefutable. The other comment is about eyes and brain. And I certainly agree with Jeff that as the survival of patients is extended, we're going to see the development of focal neurologic episodes that people fully recognize. And I think at the moment, many of these episodes go unexplained or attributed to transient ischemic attacks, which really misses the point. Getting drug to brain is critical. There are 12 mutations that are rapidly progressive and develop a leptomeningeal amyloid at early time points. And clearly that's an initial target. The challenge is, I think, collecting and applying all the lessons from the Alzheimer drug development story and applying that data and approach or multiple approaches to leptomeningeal potential in patients. These people have been left behind. People with eye disease obviously have been left behind, as has autonomic disease. So I think there is plenty of opportunity here. I do believe that central nervous system disease is being addressed by drug partnerships already. Alnylam, Regeneron, and others have clearly targeted this as the next battlefront in amyloid-related disease, even if it's not TTR. So I think we're in really good position to take advantage of all the advances. There's a lot of science that we need to grapple with, but I do believe that drug development for the CNS is a topic for now and not the future.
Thank you very much, Dr. Burke. Last, for the perspectives, I would like to recognize Dr. Ken Sakashima, who is joining us from the Pharmaceutical and Medical Devices Agency in Japan. I want to thank you for your patience. I do realize it's past 11 p.m. over where you are, so thank you so much, and we are looking forward to hearing your thoughts. Thanks.
Thank you very much for the introduction and the invitation on this great meeting. So I'm Ken Sakashima from PMDA and a medical reviewer with a background of neurology. It is important to thinking about the symptom other than the cardiac event and survival in this stage. At that time, so as Dr. Jeff Kelly mentioned, it is important to know about the manifestation of CNS symptoms and understand the natural history to thinking about the development of the drugs for CNS involvement of ATTR amyloidosis. In addition to that, so based on the understanding of CNS involvement in ATTR amyloidosis, sometimes it is required to longer evaluation for the evaluation of the CNS symptom. In that case, it is important to use or understand the current information collected at the previous clinical trials. From that point of view, so global clinical trial also is also important to conduct the clinical trial efficiency. And as I taking part in this meeting, so involving other than the United States and European countries, such as including Japan and other country, will be the good way to enhance the drug development for the next stage. Thank you.
Thank you very much, Dr. Sakashima. So for the remainder of this session, we're now going to switch into the discussion session, and I will hand it over to Dr. Preston Dunman.
Thanks, Rosalind. When you listen to the discussion that's happened here this morning, there are good things about dichotomization of disease. There have been drawbacks to dichotomization of disease. One of the realities here, though, is that coming from that place where we've done clinical development, as patients have begun to live longer, these other areas of involvement are becoming manifest, and specifically leptomeningeal involvement with this disease, as well as ocular. And it dawned on me looking at this that the answers to how to deal with this from a clinical development point of view and a regulatory point of view may be different if you consider, say, ocular involvement to be secondary to what's going on in the systemic part of your body and it's just spilling over into your eye, versus it's a primary ocular disease because your retinal pigment epithelium is actually generating TTR inside your eye. And so that may need to be a primary target. And the same I think could be applied to the choroid plexus and the CNS. And certainly the drugs that we've approved for the periphery and the nervous system really don't have anything to do with what's going on in your CNS if the drugs don't penetrate your CNS. And then obviously there the manifestations are cognition decline and bleeding into your head versus these peripheral symptoms of peripheral nerve involvement. So understanding the answers to my questions may be different depending on whether we're talking about the nervous system or the eye. Wiley, I'm going to pick on you first and start with the eye and ask you about this. People are getting these syndromes, it appears, after years of being treated systemically. When you see this in the eye, from a regulatory point of view, do you consider this a primary thing, a primary disease of the eye, or something that's just spilling over from ineffective treatment of a systemic disease, and so it's not really something that you'd really think about, say, in the ocular division of a regulatory agency?
Wiley Chambers, I'm the director of the Division of Ophthalmology. I don't know that we care. What the patients care about is they can't see. And so to the extent that you can make patients able to see or keep them from losing vision, either is an approvable indication. So we think this is straightforward to monitor. You can monitor what it decreases is visual acuity, something readily measurable throughout the world. The opacities that occur within the vitreous can be seen on B-scan, something else readily available, and those other areas, including the retina, can be observed by optical coherence tomography or OCT. So it's all measurable and all monitorable. I just don't know that is customarily being done.
Okay. I know we've got someone else from the ophthalmology division here. I think Dr. Lloyd is with us as well, Ray Lloyd. And you may be virtual, but I was wanting to ask you for your comments as well. Dr. Lloyd may not be with us. Okay. Okay. And so in thinking about this from a neurology standpoint, rare disease, natural history is pretty poorly defined, although we know this is happening after you've been on treatment in some of these patients for a long time, may have a primary source inside the central nervous system. What are your thoughts about pathways for approval of medicines for this? Because in something that takes years to happen, I mean, are people going to have to show, or developers going to have to show that you blunt this deterioration of cognition loss, or you prevent bleeding into your head, in which case these could be really long trials? Or are there potentially structural biomarkers or imaging endpoints that might be considered in a development program to treat people like this?
Hi, I'm Laura Jowidzic from the Division of Neurology 1, FDA. So these are interesting questions, but I would say we have to step back a little bit to your first point that for the CNS manifestations, because the natural history is poorly defined and mostly only defined in patients who have had liver transplant, I think the first step is to have a better understanding of the natural history in the patients who are treated with the approved products, because I can imagine that could potentially change the trajectory or affect the natural history or the time course of when these symptoms occur. The second thing that we would need from our perspective, the regulatory perspective, is clear diagnostic criteria for this syndrome, because it was mentioned that the patients have focal neurological deficits. Well, that's a very broad statement. It's a very broad category of events. So we'd have to understand, in order to enroll patients in a trial to study the CNS manifestations, exactly what are we talking about? What events, what symptoms? Because in order to have an endpoint for such a trial, you need to know what you're trying to measure, what are the symptoms that you're concerned about or capturing, and then making sure you enroll people who have that syndrome or that meet the diagnostic criteria. That I would think the community would have to come together and figure out what do you mean by the CNS manifestations of ATTR. I imagine everyone has their own idea right now, because there isn't really a set, diagnostic criteria for that. So to the next point about biomarkers and such, I think we're still a little bit far from that until we know what exactly the syndrome is and how it's defined and how you're going to enroll those patients. I'm not sure we can discuss whether imaging would be sufficient or biomarkers or things like that. It would be a bit premature for that particular entity for CNS manifestations or leptomeningeal manifestations.
And so based on those remarks, I'd be interested in the remarks of the academic community as well as industry as far as, okay, is the Boston criteria for diagnosing CAA, the cerebral amyloid angiopathy on MRI, is that good enough to use to follow as a marker to predict outcomes? And if anybody's got experience with that, because I know that scale does exist, but it's not clear to me how or whether or if it's being used in any kind of developmental way. And Sharmila, you may have some experience with that, but I'm just not aware that anybody's really published about that.
I would just comment, I think the point raised is well taken. I think there's an opportunity, as Dr. Burke and Dr. Kelly were emphasizing, the time is now for us to really start to delve into this phenotype. As we all know, patients with amyloid are very given of their time. They're all coming to our centers for various clinical trials. We're concerned none of these drugs get into the blood-brain barrier, so we have a natural history study. And I don't know, but I would imagine that many of them would participate in further defining what the phenotype is collectively so we could really figure out what the natural history is. I'm sure there are a host of concerns and complaints that have gone unmet and unrecognized. That's my perception.
Yeah, no, I think that's an excellent point. Unfortunately, I don't have much knowledge about the CAA and imaging manifestations and whether they would be similar with TTR CNS manifestations, but that's something that needs to be studied, obviously.
Yeah, I make one observation that one of our faculty made. We've seen a lot of patients who with wild-type TTR who are obviously on anticoagulation with atrial fibrillation, and a disproportionate number, we thought, or he did because he's a smart neurologist, of subdural hematomas. And you can imagine if there's rare variants that cause leptomeningeal involvement, maybe wild-type TTR has some involvement in leptomeninges. And, these are not highfalutin data, but in large statewide databases, over 10 million people, the odds ratio of having a subdural, if you had a cross-reference diagnosis of amyloid, was 10-fold higher than the control population, suggesting that, we're giving anticoagulants because they have atrial fibrillation, but there are always consequences to two sides of the coin. So,
We have an attendee today, Dr. Sylvia Kahn, who's from EMA Neurology, that I'd like to invite her to comment as well. And thank you for being up late to join us.
So I can understand you now.
Yes, we can hear you.
My name. Okay. So I'm very sorry. My name is Sylvia Kuhn. I'm a clinical assessor at the Federal Institute for Drugs and Medical Devices in Germany. And what I say here is my personal opinion, but not a harmonized position of EMA or the BfArM. And I can strongly recommend what has been said by the FDA colleagues, that it is very important to better understand the disease. We have no experience with any endpoints that would address the CNS or ocular aspects of the disease, because these have not been evaluated in the pivotal trials that were submitted for the approved polyneuropathy indication. And we need better understanding of what is really disabling patients and which aspects of these disease which appear to escape the approved treatments might be addressed by future treatments, and how to evaluate them. Thank you.
Thank you for that. That's an interesting ask from everybody who deals with this on the regulatory side to those of you who produce the information, that the natural history of this disorder is going to be important to define. In the last 2 minutes that we have here, we've got the luxury of having several people from FDA's real-world evidence and epidemiology group with us here today. And I'd like their thoughts and comments about thinking about how real-world evidence and epidemiology might be helpful and what the regulatory need would be for that kind of information, either externally controlling a trial in, say, CNS or the eye, or helping to define the population potentially at risk for rapid progression of the disease. And so I'd like Dr. Jenny Lee, if I'd start with you, if you wouldn't mind commenting on this, understanding that the FDA has put out almost 10 guidances on this here fairly recently.
Hello, thank you. My name is Jenny, last name Lee. I'm Associate Director for RWE at Office of Civilian and Epidemiology at CDER. I will comment at high level the RWE program at FDA, and my comment may not be product specific or disease specific. Very briefly, FDA was required to consider real-world evidence to support a new indication for a drug that's already approved or to satisfy post-marketing study requirements. So FDA issued RWE framework providing key consideration, how do we evaluate real-world evidence? For example, we look at whether the data source are fit for use, whether the study design can provide adequate evidence, whether the study conduct meets FDA requirements. Under this framework, we issue many RWE-related guidance. One of the guidance focused on external control trials. And there are also other guidance, for example, on data sources, EHR claims data or registry. We also have a guidance on key regulatory consideration when using real-world evidence. You could find those guidance at the FDA's website on RWE. Lastly, I want to say that FDA hold a same evidentiary standard when consider real-world evidence in support of approval. So the external control guidance is not the only guidance that mention about key consideration when sponsor could use external control trial in support of approval. For example, there is another guidance for demonstrating substantial evidence of effectiveness. That guidance also talk about the use of external controls, especially historical controls. In the beginning of this meeting, there were mentioned there are time trends of changing early diagnosis and some uncertain natural history disease progression. I think those all need to be taken into consideration when sponsors consider using external controls. I will pause here to see whether Matteo has anything to add. My colleague from OMP, thank you.
Right, thanks. Matteo Raman, Senior Epidemiologist from RW Analytics team in the Office of Medical Policy. So yeah, Dr. Lee, my colleague, explained it very well. So I would highlight a couple of points. Our recently published external control guidance in February talks about the design considerations and the best practices. One of the things highlighted in the guidance that an external control trial could be more applicable or generate more cogent results where the natural history of a study is understandable. Now, with the discussion that we have heard so far, the natural history of this disease is not very well understood. So if the community, stakeholder community can come together to gather some studies to understand the natural history better, so that would be helpful. But again, I echo Dr. Lee that the guidance specifically mentions where it is applicable, but one specific thing, all of the guidance commonly mentions that engage early with the regulatory organization to know and see if the design is appropriate from the very get-go. So I'll pause there for the sake of time. Thanks.
Thank you so much. And I've been given the okay to go just a few minutes over, so I'd like to get the comments from that may be on the minds of some of our industry colleagues. And so I'll start out with Andrew Slug, if you're here.
Okay. Thanks so much, Andrew Slug, Elm Island. And I'll just echo the need for, good quality real-world data on natural history. I mean, just as an anecdote for Apollo, we were able to use data from about 150 patients, gathered from 3 sites in 2013 to look at the change in mNIS+7 relative to their time since diagnosis to come up and model what we predicted would be the decline of placebo. It was only about 7 points off when we conducted the study. That enabled us to power a study in about 200 patients and conduct an efficient clinical trial. Now, I know that's probably not always going to be the case when you deal with CNS disorders or maybe slowly progressing ocular disorders like we have here, but it does give us, kind of a common framework from which to operate. And there's also been some excellent work, I think, done by the Foundation for Fighting Blindness and Stargardt's disease, also slowly progressing ocular disorder. And they've done some great work in developing a registry and natural history data. Perhaps there's some other diseases that we can learn from, as well as Dutch type CAA, which I heard mentioned as well. So I think a lot of good things outside of TTR amyloidosis that we can learn from. Thank you.
Great. And then Michael Roberts.
Yeah, thank you. I'm Michael Roberts from Carino. I do want to echo the statements about the natural history study. That's our primary concern or issue, because I think many of the physicians here, the centers, probably only have a few patients, right? So it's going to take a lot of work to be able to gather all that information. So that's a concern that we have, and, again, we're interested in, helping to facilitate this. So, but I think that's important, because that goes to what endpoints are we going to define in our clinical trials? Can we use surrogate endpoints to kind of bridge that gap? So there's a lot of questions that we still have.
And to your point about the sparsity of patients, I know that there's difficulties even across centers in the United States sharing data for privacy reasons. And we, this group from the Amyloidosis Research Forum has actually published a paper recently. Dr. Sinorovich was the first author on that, I think, looking at federated learning approaches to amalgamating data sets large enough to show an outcome or to show analyzing, in that case, NT-proBNP and the value of that with change of therapy. So that might be an approach that might be usable to get around some of these privacy and data sharing issues. And then last, yes, we got Michelle Campbell. Are you with us?
Hi, Kristen, and thank you so much. My only comment I wanted to add, and it really echoes what a lot of folks have been saying, and I think my colleague, Dr. Laura Drozik, who I work very closely with in the Office of Neuroscience, is we need this better understanding of these CNS manifestations, the ones that look like, and I think we have been hinting and talking about the opportunities that data sharing can offer us. And I do think, particularly when we have small numbers and small amount of information, we need to pool data together. We do need a data share. And I think I just want to use this opportunity to highlight the rare disease data analytics platform that the agency has been funding through the Critical Path Institute, because that is really the idea, is to be able to pull different study designs, types, clinical trials, observational studies, natural histories registry, to really allow us to look at the full spectrum of patients, pull them together, see what is the symptom beginning, what does it look like, how does things progress over time. I think data sharing is really going to be the one potential good solution that hopefully can come out of this to help us better understand the CNS manifestations, help us be able to kind of better understand timing, the progression. Is this a fast progressing aspect? Is this a slow progressing? And then that will really be able to drive us into figuring out what is the best way to measure. We are very well versed in providing advice to rare diseases and really thinking about appropriate study design and endpoints selection. in our rare disease clinical trials. But unless we have a really good understanding of the disease or the specific aspect of the disease, as we're speaking now in the CNS, it can make it a little bit more challenging. And so I think if we can work together and really something such as this group in the pre-competitive space, leveraging this information, will only then be able to really foster conversations when it is time to approach any regulatory agency that will be more thoughtful, because we do have this baseline aspect of what is natural history, what are we really calling this, how often is it diagnosed, and I think data sharing is going to be really this opportunity to help us better understand what is going on with some of these manifestations.
Thank you so much, Michelle. And we got one last question I'll be able to take before somebody gets a shepherd's crook after me here. So Jeff Kelly, you had a question.
Yeah, it's more of a quick comment. I wanted to just say that I think there's a tremendous opportunity here, particularly for the large amyloidosis centers, to take advantage of the advances made in cerebral amyloid angiopathy and the various dementia scores in Alzheimer's disease, that we start using these tools that are well established for Alzheimer's in the TTR patients being treated with various medications. Because I do think that it seems pretty clear already from the Portuguese studies that the various CNS symptoms are presenting more slowly in individuals treated with tafamidis. I think there's reason to believe that, based on what John Burke said, that intrathecal administration of other agents could be useful as well, particularly the mRNA lowering agents, as has been recently demonstrated for tofersen and SOD-ALS. So we might be able to get a pretty good sense of natural history just by studying patients on various medications and learning more about the extent or lack thereof of CNS penetration. So that's the message.
Okay, thank you for your comment. And with that, I will go ahead and end this session for a 5-minute break. But first, thanking our colleagues from Japan and from Europe, Sylvia and Ken, for staying up incredibly late to join us. Thank you for being here. We'll take a 5-minute break and we'll reconvene at 10:36. So, thank you all very much.
So welcome back from a quick break. It's my pleasure to introduce my colleague who will moderate the next session, Rick Ruberg from Boston Medical Center. And this is a topic near and dear to both of our hearts and many people's, racial and ethnic disparities in transthyretin amyloidosis.
Thank you so much, Matt. It's a privilege to moderate this critically important session. And without further ado, I will introduce my good friend and colleague, Michelle Kittleson, who's coming to us from Los Angeles to present the overview of this particular discussion section.
Fantastic. I'm assuming you can all hear me. I'm going to dive right in as you find your seats. Racial and ethnic disparities in cardiac amyloidosis. I have no disclosures. So to start with, let's just talk about, from this fantastic article that appeared in JACC recently, what are social determinants of health? And it's important to recognize that problems with healthcare access and quality, neighborhood environment, economic stability, education, social and community context lead both directly and indirectly to worse health and well-being outcomes. So in that context, let's talk about amyloidosis. You've heard from way bigger experts than I about what amyloidosis is, but a quick overview to frame the rest of my slides. It's a condition where a protein infiltrates the myocardium, resulting in a restrictive cardiomyopathy. The two major forms we think about are the light chains or the TTR protein. The TTR protein in turn comes from one of two sources, a mutation in the gene or wild type. It's also important, of course, as we've discussed in great detail, that it doesn't just affect the heart, it affects other tissues, which is important not only for patients, but for clinicians when they make the diagnosis. So let me give you some facts about amyloidosis and then frame these facts in the context of disparities. So fact one, in general, best case scenario, the diagnosis can be tricky because heart failure symptoms are simply the tip of the iceberg. And we have to think as clinicians about other more subtle symptoms. We know we're getting better at that based on some of the other studies you've seen, but we have to have a high index of suspicion in the face of clinical clues, including things like fatigue, other cardiac disease, neurologic symptoms, musculoskeletal symptoms. The second fact that's important to remember is that there can be more than one diagnosis, and some of the classic things we memorize in medical school don't necessarily hold true in real life. For example, classic low voltage on ECG is actually not very sensitive. Only 40% of patients will have it. Another fact, if patients with garden variety HFpEF, assumed to have HFpEF, admitted to the hospital, 13% in one study have amyloidosis, an opportunity for missed diagnosis. And in patients with severe AS sent for transcatheter aortic valve implantation, 16% have underlying amyloidosis. So we know diagnosis is tricky. It's tricky because there can often be more than one diagnosis. Another fact you need to know in the context of disparities, TTR is prevalent in Black Americans. This is a beautiful figure from a fantastic review article by Dr. Maurer and colleagues, and we know that there's a spectrum of variants with predominant neurologic versus cardiac features. The V142I mutation is present in 3.4% of Black Americans. Of course, there will be incomplete penetrance, variable expressivity, but this is a population in whom we need to have an additional high index of suspicion. Fact three, we've already heard the results of the landmark ATTR-ACT trial. But we know we have many insights to show us that earlier treatment is better. We know that in ATTR-ACT, the survival curves took 18 months to separate because this doesn't reverse disease, it just prevents progression. You have to be on it for a while to accrue a benefit. So the earlier you start it, the better the patient will do. We also know from subgroup analyses that the less sick you are at time of diagnosis, the better you'll do. So putting these facts together, we have a challenging diagnosis of a condition that's more common in Black Americans. We have risks of delayed therapies. That's basically a perfect storm for racial disparities in cardiac amyloidosis. So that's the theory behind why racial disparities might exist. Is it true in practice? Well, we know from studies that Black Americans with ATTR cardiomyopathy do worse. Now, this is a study that compared variant versus wild type disease, and the V142I variant, which was in African Americans, mortality was higher, cardiovascular hospitalizations were higher. Now, you might say to me, you know what, maybe this isn't disparity. Maybe it's biology, maybe it's a variant that's the problem, not the racial composition of the patients. Okay, well, there's another study that looked very interestingly at Black patients versus white patients with this mutation, finding a group of Italian Americans who happen to have the V142I mutation. And what did they find? If you compare Black versus white patients with the same mutation, you see these Black patients have lower blood pressure and pulse pressure, reduced kidney function, lower stroke work. So you have to wonder, were white individuals diagnosed earlier in their disease course? Do Black individuals really have more severe disease, or are they just more vulnerable to delays in diagnosis? There are other examples of disparities. If we look at 8 trials of ATTR cardiac amyloidosis by country, the darker red is more trial enrollment. We see that there's no trials enrolling on the African continent. We see that percentage of Black participants in trials is very low, though the variant is so common in Black Americans. So there's a perfect storm. There are opportunities to do better. And I would say that SCAN MP is a step in the right direction. This is by Dr. Ruberg and many of the other experts in this room putting this together, a longitudinal observational cohort study, multi-center, taking individuals who are older, Black race or Caribbean, Hispanic ethnicity with increased left ventricular wall thickness and doing screening to to determine the prevalence of ATTR-CM, the proportion of wild type versus hereditary disease, the differences based on demographics, a step towards the utility of screening programs. So what do we know? We know that it's more common than you think. We need to have a high index of suspicion in the face of clinical clues. We know this mutation is prevalent in Black Americans. We know that earlier treatment leads to better outcomes. We need to know if these differences in outcomes are related to biology and/or disparity, and is there a role for active ascertainment in at-risk populations like Black Americans? We have to remember that as we discuss the science, we can't forget the elephant in the room, that of biology and disparity, because it's not great science that saves lives. The proper equation is great science, and effective implementation equals lives saved. Thanks so much for your attention. I'm delighted to hear the discussion led by Dr. Ruberg, though my clinic starts in 13 minutes, so I'm going to have to leave early.
Thank you so much, Michelle. And yes, I was going to point out that your clinic starts in about 12 minutes now, so we appreciate you making the time for us today. This is such a critically important topic, and I think we have a unique opportunity here. Thank you to ARC for having gathered this convergence of regulatory experts, researchers in the field, clinicians, patients, and industry partners, who I think we're all aligned around the same goal, and that is to get the best treatment to the most people in an equitable manner. And diversity and disparities is a critical aspect of representing, I think, a barrier that we have not really, I think, addressed as well as we probably should have to get the best treatment to the most people. So there are two panels that will be discussing these topics today. This first panel is regarding diversity, and the second panel is more specifically around clinical trials. So many of the things that we start to talk about this morning will probably lead to this afternoon. And I'm honored to have such a great panel to begin this discussion today. Joining us remotely is Dr. Keith Ferdinand from Tulane University, and here with us in Washington, Dr. Khair Shah from Virginia Commonwealth University, and Dr. Victoria Sangiorgio from Ionis Pharmaceuticals. So I'd like to begin, kind of frame our discussion around two aspects that Michelle really nicely illustrated. The first aspect, which I think I would refer to later in our discussion, regards the biology of disease. I think that's a little bit out of scope in some ways for the bigger discussion of this first topic, which is really about addressing disparities and how to basically overcome those barriers. The first barrier, I think, really involves awareness. And I'd like to first ask Dr. Ferdinand and bring him in from New Orleans. So Keith, how do we raise awareness of ATTR amyloidosis in the African-American community, particularly around the complex issue of hereditary amyloidosis? Thank you.
Thank you for this opportunity. If you take into account there are approximately 47 million persons who self-identify as Black in the United States, with a penetrance of 3.5% of the allele, that's about 1.7 million people. That's a lot. But you also have to remember that the overwhelming cause of heart failure in the Black population in the United States is untreated hypertension. So it gets very complex, and I often ponder whether we should spend all of our information simply on one form of heart failure, perhaps teach our patients that if they have fatigue, shortness of breath, or other symptoms with a positive family history and those red flags, to think of heart failure, and then perhaps have our primary care practitioners and our general cardiologists make the diagnosis, put it together. It's going to be really hard for patients just by simply doing an awareness campaign to put these things together in a package and come with the diagnosis when the physicians and other advanced clinicians aren't doing the job. I put in the chat also, the plot thickens. There's more hospitalization, more death related to heart failure in general, more HFpEF. So perhaps if we just treat our patients in an appropriate manner, we're going to get those diagnostic facilities available so that we can make the diagnosis of amyloid cardiomyopathy. I'm not really sure just going to the general population and doing that is the way. The other problem is if you overscreen, you're going to lose some of the sensitivity in testing. So we have to look at the ability to not overdiagnose. And I think that was mentioned earlier today. Those persons who are getting technetium scans and they're being misinterpreted, it can lead to a pathway of using very costly, complex interventions when they're not needed. So it's not an easy thing, and it's not enough for us just to say we're going to educate all Black Americans about amyloid cardiomyopathy?
Thank you so much. The critical points, and I think your point is obviously really well taken regarding the important but smaller role that this particular disease plays in the larger burden of heart failure in Black patients in the United States. So Kara, I'd like you to share your thoughts here. Where do you think are potential opportunities for us to kind of increase this awareness gap?
Thank you very much, Dr. Ruberg. And for folks in the room I've not met before, I'm a clinical cardiologist at VCU in Richmond. And I'll start by following Dr. Ferdinand's comments. I think the conversation for this disease is even more complex than just race or ethnicity. It's not just Black patients. It's elderly Black patients. It's elderly Black patients with a potentially genetic disease that may affect their family members. And in the United States, it's elderly Black patients with a genetic disease that are more concentrated in the South and Southeast, where traditionally outcomes with cardiovascular disease have been poor, access to clinical care for these populations have been poor. And in Richmond specifically, it's almost a case study in racially segregated neighborhoods. Arising from disenfranchisement, redlining. VCU is centered right next to I-95, which goes right through traditionally Black neighborhoods. And classically, at least in Richmond, these patients have been underserved and distrustful of medical care. So it's a much more delicate and challenging topic than I think just hypertension or traditional medical diseases, which maybe are singular in characteristics in why patients have poor access. What we're working on, and it may not be applicable to most, but I feel like most clinicians in the U.S. that treat amyloid are treating a population similar to me, is we're working to work with local federally qualified health centers and community health centers. And we find that these clinics traditionally have done well with other disease processes, in that the members that deliver care, that govern the clinics, that are patient interacting, are actually members of the community, and they're invested in the population, and they seem to have a much better trust and relationship than some of the specialized centers at the university hospital. So we're hoping to report on in the future on whether this is an efficacious intervention. But I do think, I do appreciate the interventions from a policy and systemic and institutional basis, but I do think a lot of the success that's going to be gained in really making progress here is going to be on the communication, patient interaction, and sort of boots on the ground perspective.
Thank you very much, Dr. Shah. Did you want to make a comment?
Yeah, I just want to make a quick comment. When we looked at our data of African Americans with ATTR cardiac amyloidosis, 28% had wild type. And there was about 53 patients in that cohort who had wild type. So, we often talk about African Americans at risk due to the V122I mutation, but elderly African Americans are at risk of wild type as well. So just something to keep in mind.
Yes, I echo those points. We're about to publish very similar findings, so definitely, I think, underappreciated. So, Dr. Sanjurjo, Dr. Shah has actually anticipated a number of the points I was going to make, and I'd like you to kind of maybe pick up on those if you could. And that has to do with kind of dissemination. What is the participation of sponsors in terms of dissemination of information to communities so that we basically kind of not only enhance awareness but also kind of engage trust? I think which is kind of a critical aspect to enrollment.
So just to introduce myself a little, I'm in the clinical development team at Ionis, and my passion and focus for the CardioTransform study was to make sure that we had enough Blacks and Hispanics in our study. I am a Hispanic physician, so there's not many of us in industry that sit at the table when we're making decisions or looking at the protocols and picking, what sites are going to be selected, et cetera. Currently, right now, I know that this came up also. The problem is that clinical trials in general do not go to where these patients are. We always pick the large institutions. So working with community-based hospitals is number one place to go. That's where the patients are.
Right.
Also, speaking of, I think I wanted to bring up a point that Dr. Ferdinand mentioned regarding knowing that you have a family history. That's a problem with the Black and Hispanic community. They are not being tested to date. So everyone says, you know, Hispanics and Blacks do not like doing genetic screening. Well, there's a paper that came out that showed, you know, we have a poster where 22,000 patients were sent out for Ambry testing. Large population were Hispanic and Blacks. So if you have free genetic testing, you see that they will be tested. So I think awareness like that is very important because these Black patients did not know that they had a family history. Right. So you can ask all you want, but if they don't know, you know, I think it's important that education, as far as industry is concerned, we've done a poor job up to date of reaching out to the communities. I've been working in diversity for 20 years, not officially, because I'm Hispanic. I want to make sure that I represent. Now, luckily, with the DEPICT Act and with, you know, the FDA guidance, this is a game changer for all of us. You know, I'm very happy that we had a diversity plan, very comprehensive, before, you know, it was mandated to do it. And I know all of our investigators would hear from me all the time. You know, I think they were tired of hearing from me, you know, guilting them to see where, you know, our patients were. So, you know, we're moving forward and we're improving, but industry to date, we do have to reach out to communities and start working with them. And community hospitals are number one because Black patients tend to do better with Black physicians. Latest.
I really appreciate your comments and your candid comments. And actually, I would like to invite other members in the room to share their thoughts on this matter, specifically involving, I think, trial design such that more active engagement, not with the academic medical centers, or at least academic medical centers that have formed partnership with communities, which I think is one potentially way to go, as well as again dissemination and communication of information regarding the intervention and also the disease itself. And genetic testing, I'd say, for another part of this discussion, because I think that's also critically important and something that Dr. Maurer and I have thought a lot about recently. Anybody? I'll open up to everybody. Any comments? Oh, you have your hand up.
Yeah. I've been thinking about one way we can harvest more patients and get them more available for early diagnosis, since the tafamidis has been shown to be beneficial in those patients early on in disease, is by a test that's used omnipresent even in community centers and private doctors' offices. That's the echocardiography. And many times they're making a diagnosis of LVH. We know it's not true LVH, but increased wall thickness. They're not recognizing discordant ECG pattern with it. Those are some patients who are already getting one of the early tools that may lead to appropriate diagnosis, but I'm not sure that the community cardiologists or the primary care clinicians are aware of that particular pathway. So that's something we may want to do, is highlight in community centers, in private doctors' offices, in private practices, the need to be more aware that what we're calling LVH is increased wall thickness, and if it's discordant, especially with, and I agree it's often difficult to document, but especially with that family history and other red flags, that we're missing those patients. We're seeing them, they're there. One more point, Dr. Alexander out of Stanford did a heat map, and it seems that there's less amyloid cardiomyopathy death in the South. Well, that doesn't make sense, because in most of the southern states, I'm in Louisiana, it's the legacy of slavery, there's anywhere from 25 to 35% of the population would be identified as Black. So how can you have less amyloid? Well, there are less centers there, there's less diagnosis. The pipeline is not being entered for these patients to get diagnosis. So that's one of the things we can do, is highlight for those clinicians who are doing a test which may assist along with another test that's omnipresent, the ECG, but they're not looking at it appropriately.
Thank you. Would you like to make a comment?
Yeah, just, I guess a couple of comments here is that, again, just forgot to introduce myself. I'm Adrian Hernandez, a cardiologist at Duke and director of the Duke Clinical Research Institute. If we really want to try to reach out to diverse communities, we will have to do something very different than what we're currently doing, because I'll just use Duke as an example. And so while our medical school has had significant improvement in terms of diversity of our class, in the most recent classes, over a third in terms of diverse backgrounds, if you look at the timeline for that, for them to hit practice or academic centers, that's a long way off. So you're talking about 10 years. Now, what we do have and have had great success is bringing in other disciplines in healthcare. So nurse practitioners, physician assistants, clinical pharmacists, and they are very motivated to engage in community-based research and also serving populations that have not traditionally been reached before. And so I think, opening up the aperture a bit in terms of the disciplines that come in here is important. And then I guess the second thing, which ties into this complex issue about awareness. And so, as commented earlier, there is, remains still distrust in the community. And so, like, you can imagine scenarios like, hey, we're going to go out and screen for people who may have amyloid and also to offer genetic testing because that could actually have an impact. However, in the past, that has not always been well received unless it's done with the idea to do something after just having those findings. And so what's going to be the commitment to the communities?
Thank you very much. Those are critically important points. Thank you very much for making those. I'd like to echo one of the points that you made, Dr. Hernandez, and that's to do with kind of community engagement in terms of research practices. And I'm curious to know what the group thinks about those sorts of activities. An example might be forming community advisory boards to provide feedback on, and maybe not so much trial design, but on trial kind of productivity and the actual kind of comportment of a trial. And I'm curious, do people have thoughts on that or have experience with that? That's something that Dr. Maurer and I have instituted in our study, which I think has been very, we've learned a huge amount from, and I think has been very impactful to the study.
We're doing that here. Yeah, we're doing that here. Excuse me, there's a delay.
Just comment that, someone on our community advisory board whose father passed away from amyloid from V122I, who's an allele carrier, has no disease, she felt like this should be apropos of other screening approaches, that African Americans should be aware that at the age of X, 50, 60, that they may want to get genotyped, and there should be some, broader campaigns. To Keith's point, I personally, my experience is that the key here is trust. And I think we're missing an opportunity, that is, at the amyloid centers, when we've diagnosed someone who has V122I and we've taken care of them and now they trust us. We have an opportunity with this disease, which I think is unique among genetic cardiomyopathies. It's unique, because we don't have variants of unknown significance. We order a TTR gene test and we know you either have it or you don't. There aren't these, I don't know what to do with this, or a variant of unknown significance, or a VUS. The dilemma that's also different is we don't need to really screen I think at this point, their kids. We really need to screen their, brothers and sisters who are the age, because it has age-dependent penetrance. So, I don't know, we have this cuckoo idea Rick and I have been working on. I think we can also learn a lot from, God forbid, the COVID pandemic, where we can leverage technologies. We're looking at people all over the world now on Zoom. But these patients, as you pointed out, Keith, don't have access to expert care in amyloid where they're living. And I think it's behooving to the amyloid experts to try to figure out a way to engage them and make sure that they get adequately tested. For pharma, I would say you want to screen individuals and find a 5% chance of having disease or a 7% chance with carpal tunnel or other things. This is a 50% chance of having individuals who could be affected at this particular point in time. So I think it's all lined up to do the right things. We just have to think outside the box of how to approach these individuals with, I think, as Michelle was saying, experts from implementation science, because none of us are, certainly I'm not in that realm, to really better advocate for this population.
Thank you very much, Matt. No,
Can I just jump in and say,
please.
I think, it worries me when I hear it's driven by research. I think first and foremost, we have to support and better treat a community. We have to get rid of that disparity. patients who are well cared for, who are well educated, are more likely to participate in research. And I think that, as others have touched on, but that's the absolute driver, is that we have to take the care to the patients. It shouldn't be that patients have to travel for care. I think we have to rethink how we care for patients across different geographies, across different socioeconomic and racial backgrounds. And I think we do a poor job of that. And often the conversations are driven by research, but it has to be driven by care and by needs.
Thank you very much for that excellent point. I would make one point, then Angela, I'll pick you up in just 1 second. And I would argue that it is truly an equity issue. By not providing our patients access in a fair manner, then we're depriving them of fair care. And I think that is kind of it's antithetical to what we're trying to do. So, Dr. Dispensieri, please.
Hi, Angela Dispensieri, Mayo Clinic hematologist. The point was made that a lot of times the patients aren't getting care at these academic centers, and it brings up a challenge in terms of trial design, because you want to have these complex trials so we can get a lot of information, but it's like a day of testing. It has to be at a special place with special stuff, and so that makes it very challenging. And so it's that compromise. Do you have a trial that can be done more in a community setting? Do we have adequate measures so that the study and the research drugs, the innovative therapies, can go to the patients where they are? I don't have an answer, but they're sort of running against each other, but we tend to make very complex trials. And I think we need to, because we're early in the stage of understanding, how do you actually stratify your patients? The biomarkers in the AL world, it's almost 20 years since we've been using that as a staging, and it really is effective in helping stratify. Do we have the confidence with staging systems in, ATTR? So just food for thought.
No, excellent points, and I really appreciate that. I think what you're basically getting at is the same thing that Isabel said, which is we really need to take kind of a more patient-centered approach when designing these trials. And I think Dr. Shah's excellent point regarding, and I'll get you 1 second, Victoria, is about most of our patients are elderly with ATTR, and so the experience that they have and the burden is even more magnified, because it's challenging for all the assessments. And I know that Matt and I thought a lot about this as we were designing our trial. How many questionnaires can we ask one person to subject them to, and how many different imaging tests can we do? So it becomes, I think these are really important, especially as we want to bring more people into trials. Please, Victoria.
I think something that hasn't been brought up so far that I think is very important is unconscious bias. 60% of the physicians in the United States are white. Less than 5% are Black and Hispanic. We need to make sure that we're training physicians, or at least they understand what.
Can you speak into the mic?
Oh, sorry. Can you hear me? Oh. I think unconscious bias training is very important. Also, ensuring equitable and fair compensation for patients, subjects, is, I think we're going to be speaking about that later on. I know people have looked into this. It's not the same. If you ask my parent-in-laws to go on a clinical trial, they're in their 70s, they work. They're not going to, say, $50 or $100 that you pay a patient to go into a study all day. It's not going to go for them. They need to work. So I think that should also be looked into to make sure that we're, narrowing down the disparities, different groups getting involved in studies.
Thank you for making those really important points. I think unconscious bias, implicit bias is something that we all, should be part of our medical training in general, and that's potentially could be brought in as part of onboarding for research study personnel. And furthermore, I think making compensations and also making it possible for participants to participate in trials financially and making it worth their time, I think, is also critically important. That is a perfect segue, into our afternoon discussion, and maybe we'll pick up that point. But I think we've reached the end of our time. Oh, there's one. I'm sorry. So, okay, we'll take one last question. Thank you. Go ahead, Barry.
I'm not sure if this is on. Is it on? Okay, great. So, Barry Trachtenberg, cardiologist from Houston. So, I think on the one hand, it's really knowledge is power, and I think it's really important that we get out to the community and and educate the community about this disease and the fact that there's so many people at risk for it. On the other hand, I give a lot of amyloid talks to very smart physicians, and it's a complicated disease. And even maybe it's on me, but I give a talk and I try to make it very simple. And then a week or two later, it's what test do I order and how do I make this diagnosis? And ordering they've ordered PYPs without doing any other workup. So I just think we have to be very careful and thoughtful about how we get out to the communities. And it's not just about educating and genetic testing on everyone. We have to figure out how to follow that up with screening algorithms that patients are actually able to get to and get to the sites that are able to do that, and whether that's industry helping to help pay for some of the testing. I'm not sure what the answer is, but just putting that out there for thought.
Rick, I can say what I have to say in less than a minute.
Okay, you got it, Keith.
You mentioned the community advisory boards, and we didn't really flesh that out. We don't have time, but that's an integral part of doing any research. Here at Tulane, we have a CHERISH study. It's sponsored by NHLBI. We're going out to 42 churches, and the community advisory board is very powerful and a big part of the study, going to those churches, telling them what the study is for, saying we're going to reimburse them for time and travel, letting them know this is not Tuskegee, where 400 Black men who had syphilis were observed by the U.S. Public Health Service and were not treated with penicillin even when it became available. The mistrust, the distrust is high. The white-Black mortality gap has been persistent for decades, and we're not going to overcome it just by going out to the community from academic centers. We have to have those community advisory boards, those trusted messengers, part of any research or interventions we're doing.
Thank you, Keith. Thank you for bringing it back to trust, which I think is the fundamental kind of link across all these discussions. So I think with that, we'll close this session. Thank you all for a great discussion, and I look forward to the afternoon.
Thank you, Rick. And I am Isabel Lusada. I'm the CEO of ARC, and I'm a patient myself with AL amyloidosis. So I am really delighted to be moderating the next session on patient perspectives with a focus on today's unmet needs. Unless we understand what patients want, we can't develop the treatments for them that they really want and need. And I think that's absolutely critical. We have heard over time from patients, and I mentioned earlier in 2015, we had one of the first to hold an externally led patient-focused drug development meeting. And I was reminded by Preston earlier that some of the symptoms that were most devastating to patients were symptoms that weren't being measured and regulators and others really didn't know about or consider. So it's really vital that we hear this. We've got a wonderful group of patients speaking today who really cover the spectrum of both ATTR, hereditary ATTR, and wild type. And so we're going to first start, though, with a presentation by Kristen Su from ARC and about the ARC's ATTR patient burden survey. So, Kristen, can I hand over to you?
Yeah, absolutely. So thank you, everyone, for giving me the opportunity to speak. As Isabel mentioned, in anticipation of this meeting, we started earlier this year looking at ways to incorporate the patient perspective directly into this discussion. And so knowing that it's been 8 years since that patient-focused drug development meeting that we held in Silver Spring, so much has changed. We wanted to be able to hear from the patient community what their experience living with TTR amyloidosis is today in the context or the presence of a multiple treatment landscape, and ultimately better understand what that unmet need is. And so I'll start by presenting the results for a survey that we recently launched earlier this year, and then we'll hear from the perspectives from 9 different patients across the TTR amyloidosis community. And so earlier this year, we designed a survey with the goal of assessing the burden of disease in TTR amyloidosis on patients and their caregivers in the setting of new therapies. This survey was conducted in just under 8 weeks earlier this spring and was available to adult patients and adult caregivers of patients diagnosed with TTR amyloidosis. For this presentation, I'm going to focus on the results of the patients rather than the caregivers. This was a global survey with translations available in 6 different languages and was recruited primarily through the patient network with ARC, with the amyloidosis support groups in the U.S., and then several different country-specific networks. We collected basic demographic information, disease characteristics, and information about patient experience with treatment, health-related quality of life, and healthcare resource utilization. Now, altogether, 390 patients participated in this survey across 16 different countries. The vast majority of the patients took this survey were in North America, the United States specifically, followed by Germany with 7% and Canada 5%. This was the first time we've been able to offer translations of our surveys in recent years, so we're hoping to increase global participation in these types of studies moving forward. The majority of patients were male, white, college-educated, and retired. The group was slightly more wild type, 56% compared to hereditary or variant TTR, and the average time since diagnosis was just over 4 years. You can see on the right side of the screen that we looked at patients based off of whether they had cardiac involvement, nervous system involvement, or a mixed phenotype. And so about 80% of the patients reported having cardiac involvement, and just about half of those also reported having nervous system involvement, so really a mixed phenotype of patients. There were 19% of patients total who reported having no cardiac involvement, and so those were patients who reported either having exclusively nervous system involvement or involvement of other organs, which we'll look on here at the next slide. Now, we've heard a lot about TTR being, and we think a lot about TTR as being dichotomized between cardiac involvement or nervous system involvement, but we know, and we heard earlier in the day, that the two don't occur in isolation. And you see that roughly half of the patients who participated in this study who had cardiac involvement also reported having nerve involvement as well. Patients reported also commonly experiencing GI symptoms, GI involvement, and to a lesser extent, kidney involvement, ocular involvement, and CNS involvement. On the far right, you see patients grouped within the no cardiac or no nervous system group, and those typically were patients who reported that they either had no organ involvement or they weren't sure what their organ involvement was, and it was a small group. This is a busy slide looking at disease characteristics, but a few points to point out here. You can see the vast majority of the patients in the group that reported having cardiac and nervous system involvement are wild type patients, whereas 42% of those in the mixed phenotype group, the cardiac and the nervous system group, were wild type as well. Of note, just under a third of the patients who participated in this study who were wild-type patients also reported having both cardiac and nervous system involvement. Conversely, those that are in the nerve-only group are more likely to be ATTR variant patients with V30M being the most commonly reported mutation present within that group. The majority of the patients were reported either being enrolled in a clinical trial or having access to and currently on commercial therapy. And about 18% of the patients in the mixed group, the cardiac and the nerve group, did report being on multiple therapies. And so a smaller but not insignificant group of patients are being able to access combination therapy across different various centers. And then last to note, the majority of those who've been on commercial therapy have been on so for at least a year. So this was the group of patients that we got as part of the study are largely being treated. One thing we did ask patients was a simple yes/no question, whether or not they agreed to different aspects about their treatment experience. So really wanting to know from a patient perspective, do you feel like your treatment is working for you? Is it slowing your disease progression? Is it stopping your disease progression, helping with symptoms, or are you not able to tell what's happening? And just about half of the patients across all groups reported not being able to tell whether their treatment was actually impacting their disease. And I can say we anecdotally asked patients through open text if there was anything else they wanted us to know as part of the survey, and the vast majority of patients reported that they really weren't able to tell whether their treatment was doing anything for them. They weren't feeling worse, but they couldn't actually tell whether they were making progress or if the treatment was helping them. Now, one of the goals of this survey was to assess the burden of disease on patients with TTR amyloidosis through questions related to their symptoms and the impacts that those symptoms are having across different aspects of health-related quality of life, specifically their daily activities, their role functioning, emotional functioning, emotional functioning, and then physical functioning. We measured this across patients using the Transthyretin Amyloidosis Quality of Life Questionnaire, or the ATTR-QOL for short. this is a PRO measure that ARC developed in partnership with Quality Metric, specifically designed for patients with TTR amyloidosis, regardless of their subtype or their symptomatology. We're in the process of validating this tool now, but we have strong evidence of content validity, across patients with varied symptomatology, and the initial psychometric evidence supporting the tool shows that it's a reliable and valid instrument for use in TTR amyloidosis. It's a PRO that's split into 2 modules covering both symptoms and impacts, and the average completion time that we've seen so far across patients is about 12 minutes. Now, patients are initially asked about the occurrence or the frequency of 32 individual symptoms and their severity. On average, across all patients, patients reported about seeing an average of 15.6 out of the 32 symptoms at some level of frequency and severity, and this was highest in the group of patients who had both cardiac and nervous system involvement. The frequency and severity of the symptoms are then taken into account to generate a symptom level composite score, which ranges from 0 to 6, 6 representing the most frequent and the most severe symptoms, so a worse symptom experience. Now here you can see the entire system linked, so the high symptom level composite score is defined as a score of 4 or higher. So on a scale of 0 to 6, 4 or higher is considered a high symptom experience. This list here, you can see the entire list ranked based off of the percentage of patients who scored at least a 4 for each symptom level's composite score, so experienced worse symptoms. The average number of high-scoring symptoms in the past 4 weeks was about 5.9 across all patients, and the highest being in those who reported nerve involvement, regardless of whether it was with or without cardiac involvement. So this slide highlights the cardiomyopathy symptoms, so symptoms that have to do with the cardiac. You can see that the symptom group that was most commonly reported across patients from a cardiac perspective, or I'm sorry, I'm having a hard time seeing this with the screen. Fatigue was the highest across the cardiac symptoms, whereas chest pain was the lowest. This next slide, the orange, highlights the symptoms from the nervous system involvement, and so the highest, most commonly reported was pain, numbness, or tingling in the feet or legs, and 4 out of the 6 of the most commonly reported symptoms across all patients were symptoms of nervous system involvement. This slide looks at symptoms from autonomic nervous system, and so sexual dysfunction was reported as was the highest reported symptom across all patients, and then followed by GI symptoms largely. And then the black bars here look at other symptoms, so symptoms like skin issues, blurred vision, headaches, and then blood in the urine or urinary tract infections. The patients are then asked a series of questions about how their TTR amyloidosis impacts their quality of life. This generates 4 impact subscale scores across subdomains of daily activities, role functioning in a family and social environment, emotional functioning, and then physical functioning. Scores range from 0 to 100, higher scores indicating a greater impact from TTR amyloidosis. You can see on the left-hand side of the screen that the nerve involvement was associated with a higher impact across patients. Patients with both cardiac and nervous system involvement were most impacted, followed by those with nervous system and no cardiac involvement. And then looking at the specific impacts across each of the subdomains on the right side of the screen, TTR amyloidosis has impacted patients' emotional functioning the most, followed by their role functioning through family and social functioning, physical functioning, and then daily activities. And then lastly, we wanted to look at health. We did ask patients about healthcare resource utilization and out-of-pocket expenses. The total healthcare resource utilization was calculated as the sum of all doctor's visits, ER visits, hospital admissions, procedures, and in-home services within the past 6-month period. This was highest in patients, again, who reported both cardiac and nervous system involvement. Patients reported a wide range of total out-of-pocket costs. Truly, it was sporadically reported across patients. I'd say the cardiac and the nervous system group reported the highest percent of those having to pay at least $2,000 over the past 3 months. But even when you look at some of the lower categories, again, remembering that this is an older population, largely retired on fixed incomes, these are not insignificant costs across this patient group. And so, to wrap up, what we saw in this survey is that ATTR amyloidosis is truly a multisystemic disease, most commonly impacting the heart and the nervous system. The majority of patients reported being on one of the four approved therapies in TTR amyloidosis, and then to a smaller extent, taking part in a clinical trial. Despite that, about half of patients feel that they don't know or they can't tell if their treatment is impacting their disease, and they're continuing to experience multiple impactful symptoms. Across different domains of life, TTR amyloidosis has impacted patients' emotional functioning the most, followed by their role functioning, physical functioning, and daily activities. And patients, again, within that reported having nervous system involvement are those that are most impacted by the disease. And then lastly, the healthcare resource utilization is the highest for those who have both cardiac and nerve involvement, with most patients reporting seeing multiple doctors and having multiple procedures related to their TTR amyloidosis over the last 6 months. And so with that, that was a look at the patients that we surveyed across the community broadly. I'll turn it back over to Isabel to hear from some of the patients that we have in the room presently, online through Zoom, and some videos that we have recorded over the past several weeks.
Excellent. Thank you, Kristen. So it's my absolute pleasure to introduce our patient speakers. And Mike, I would love you to take us away.
Okay. Sorry. My name is Mike Lane. I'm from Kansas City. I'd like to thank ARC and the FDA for giving us patients a platform to be heard. We sit here and listen as you discuss data and present graphs about quality of life and life expectancies. What you call data, in most cases, is what we, the patients, call life and death. I have ATTR, hereditary cardiac amyloidosis, the T60 mutation. I was diagnosed in 2018 by Dr. Brett Sperry, who my being here today is a tribute to his care. At that time, we thought it was heart and nerve involvement only. After being part of the University of Tennessee's PET imaging study, it also showed my amyloid was in my lungs and my spine. In 2018, when I was diagnosed, I was barely functioning. The 6-minute walk test was a marathon that I couldn't finish. From my car to the doctor's office required me to stop and rest. At that time, there were no FDA-approved treatments, and the outlook was kind of grim. I was lucky enough, after making cold calls to Pfizer, to find a doctor at Pfizer who took the time to guide me to the compassionate use of tafamidis. Eventually, a pacemaker was installed, along with other medications for symptom management. After that, Onpattro was approved, and then Amvuttra, and I was able to add that to my treatment plan. All these things helped my quality of life, but nothing could help the damage that was already done. I am on or have been on 3 of the 4 now approved medications. The progression has slowed, but my life is affected. Walking upstairs can be exhausting. If I lift anything heavy, I get exerted, short of breath. I must avoid things in life most people can easily enjoy. When I go to the grocery store, it takes everything I have to walk up and down the aisle to shop. I have to miss going to see my world champion Kansas City Chiefs because the stadium and the parking lot is just too much walking for me. I can't manage that much. My heart walls have thickened, and I would love to see some medicine or treatment that could repair that damage. I'm also living with neuropathy and coughing. Hands and fingers still get numb and tingle, and when this happens, I have to stop and let it pass before I can effectively use my hands. If I stand for any long period of time, my legs, especially my thighs, go numb. I've had some back pain, and now that I can see the amyloid has built up in my spine. I deal daily with leg swelling and recently had to add another diuretic to my treatment plan. As many of you know, that's not really a good sign. I feel this is because although we stabilize the protein, the already damaged nerves never have a way to repair. My scan showed me that the deposit levels seem to have remained stable, but there remains a significant amount of amyloid that continues to affect my normal functions. We need medications or treatments that will deplete the amyloid and give our body a chance to recover. This is the next step me and I'm sure every other patient prays for. This shows we need for even more effective treatments beyond stabilizing. Right now, we patients have nothing. I have a son who is 46 who is carrying the gene. He's being monitored with no symptoms yet. Why do we have to wait for symptoms which come after organ damage to give patients treatment? My son lives with the uncertainty of the future. No one can tell him if he will get amyloidosis. He and others will be waiting years before they know if amyloidosis will affect their bodies. With current treatment, this wait will only be over once his body has damage and symptoms appear. This is a difficult wait for all carriers. Even now, we can see the accurate diagnosis of active amyloid can be challenging. As patients, we desperately need a way to get an earlier diagnosis. I understand the need for regulation and trials. However, I also know that in my amyloid community, we have a different risk-benefit equation. We don't always have the time for traditional trials and endpoints. Many of the people in this room are working very hard to find new treatments and new ways to diagnose. As a patient, I am hopeful that this new research will bear fruit and quickly be put into use, helping all of us and our families who so badly need these things. We need much more awareness for patients and for doctors. We need better diagnostic tools. We need better treatments that can remove amyloid and reverse the damage. In some instances, we need better doctors. We need these things sooner, not later. Thank you.
Thank you, Mike. You are an amazing advocate for amyloidosis, and thank you so much for sharing your story. I'm delighted to introduce Amy next to speak. She was one of our speakers in the 2015 meeting, so it's amazing to have you back here, and let me hand over to you.
Well, I'm glad to be back and to be able to be back, so thank you and everyone in the room. How do you catch a unique animal? Unique up on it. How do you catch a wild, unique animal? Same way, unique up on it. My name is Amy. That's also the nickname of the disease that has brought us all together. I was diagnosed with ATTR amyloidosis in November of 2014 after being misdiagnosed for a couple of years and searching even before that for some explanation of what seemed to be unrelated symptoms. My variant is Phe44-Sur, and there were only 2 other known cases in the world, and those patients had passed away. Each presented completely differently. My parents would test negative, as well as my fraternal twin sister. There were no treatments, and I would die within 5 years. I had a daughter who was 8 and a son who was 12. I'm most likely a spontaneous mutation, an X-woman. Truly a unique animal indeed. I didn't have a subset of symptoms. I had most known symptoms and some hardly known. Peripheral and autonomic neuropathy, feet, burns, bruises, wounds, legs, nerve pain, falls, balance issues, hands, trigger fingers, Raynaud's syndrome, torso. Eyes, retina, cornea, glaucoma, 3 vitrectomies, cataract surgeries, glaucoma, laser surgeries, glaucoma drain devices implanted, and several issues with corneal erosion and other complications. Ears, rapid and severe hearing loss. That can't technically be attributed to hATTR without autopsy, and I'm not that curious. Orthostatic blood pressure, syncope with injuries, cardiac involvement, congestive heart failure, neurogenic bladder, removal of that bladder and reconstructed neobladder, October 2020. Kidney, chronic kidney disease, GI, stomach, bowel with feeding tube, 2015 to 2017, bowel resection, 2017, Indiana pouch, 2020, malnutrition, severe weight loss, etc. Possible central nervous system known to my variant, leptomeningeal involvement. My timing of diagnosis, while treacherous in the damage that had been done, also made me eligible for the Apollo study and was divine in slowing down the progression and flipping this disease on its head quickly. I'm on a silencer and a stabilizer. They have clearly extended my life and given me more time with my children, and will have made a difference in how my children might experience the disease if it should get passed down. While the treatments slow the progression, they are not a cure and are limited in how they can be effective, mostly focusing on the amyloid that is created from the TTR made in the liver. My symptoms suggest that some of the TTR that affects me is made elsewhere, either in the central nervous system or in the eye, where most of the treatments cannot access. It is believed that tafamidis can and does cross the blood-brain barrier, but it is clear that none of the treatments cross the blood-eye barrier, and I struggle daily with retinal issues, corneal neuropathy, and very damaging glaucoma. My independence is being threatened, and my creative and professional work more and more limited. The lack of investigation into hearing loss and the auditory nerve make involvement speculative and limits treatment to hearing aids that are insufficient and not covered by insurance. Living longer is creating circumstances that are exposing more patients to these sorts of symptoms. A new definition of the cost of living. In addition, the advice concerning children has not changed since treatment has become available. My son is now 21, but it is still not recommended that he get tested until and unless he is symptomatic, as treatment is not given prophylactically, and no treatment would be available for my 17-year-old daughter, and if either were positive, they would not qualify for life insurance, and a positive test result would most likely affect them negatively psychologically. I think more research in the field of onset of disease is needed. The assumption that most people experience peripheral symptoms first may not be accurate, as some people get autonomic symptoms before peripheral, which would rarely result in early diagnosis of the disease. So, how do you catch a wild, unique animal? It takes this very village to tame such a beast. So thank you all.
Thank you so much, Amy, and it's just a testament to what a warrior you are. We hear about the multiple symptoms that affect patients, but it really, you speak to the absolute burden and what you have to live with daily, and I think it's very sobering to hear from patients and to hear from you. We're now going to move to a couple of videos, so hopefully we can get those playing. The first patient we're going to hear from is Tina, and then from Thomas.
Good morning. My name is Tina McCrae, and I have transthyretin amyloidosis. That's a tough one to say, but it's also known as TTR, and it does affect my heart. So I have a cardiac condition, if you will. Thank goodness, I don't have any other organ involvement other than my heart. But I was diagnosed, I want to say, about 5 years ago. Well, the treatment process benefits for me. First and foremost, I'd like to share with you that at least 3 to 4 times a week, I'm up in the morning on the track walking. Now, for me, I do a walk. It is not necessarily a brisk walk, but I do have a very decent exercise plan in place. And that exercise plan has allowed me to be able to continue to feel good, along with certain medications are involved that I do take, but I have the opportunity to exercise. When I finish my exercise, I don't feel like I'm getting ready to just drop dead. I feel invigorated. I feel my body feels encouraged. So subsequently, I'm able to move out. When I say move out, do the things I want to do with my family and with my friends. And of course, as a woman in a household, there's always things to do at home. So I'm not lying around feeling threatened that I can't go forward with those kinds of activities. So what am I saying? Most definitely, I don't feel that my lifestyle has really been kind of made complicated. I have a very decent lifestyle, getting around with the grandchildren, still making those very special meals from time to time. But realistically, I don't cook as much as I used to. But I'm still able, when I get those special requests for a family dinner, I do all of those things. I'm involved in the community. I sit on the Harlem Week Children's Festival Board. I've been doing that for the past 15 years almost, and I'm still able to do it in August, one of the hottest months of the year. Now, I'm not Supergirl. I have had to learn to pace myself. My rest is very important. I get good quality rest, sleep at night. So the word that I was searching for in my head was compromise. I really don't feel compromised, and that's really important. It's a huge benefit. Initially, being diagnosed was a pretty scary or very uncertain time for me. The first thing that I think that your team may want to give strong consideration to is the fear factor. Someone's just been told that they have a serious illness that affects vital organs of the body. Because you can have all of the knowledge in the world as the technician, the doctor, so to speak, but if you can't convey that to the patient, and if you don't have a patient, you're nowhere. Let me just say this. You want to make sure the patient is aware there's an excellent opportunity for a better tomorrow, that this is not the end of the road. There are excellent opportunities for a better tomorrow with the type of patient care that you can expect to receive. And I would just like to say, by going on record and saying, thank you. Thank you, Dr. Maurer. Thank you, Columbia Presbyterian, for giving me the opportunity to let folks know how good you are.
My name is Tom Ross. ATTR. I have ATTR amyloidosis. And the year that I was diagnosed was 2021. The organ impacts for me are largely cardiac. I think that the PET scan indicated like very low accumulations in other areas. But for me, it's mostly cardiac amyloidosis in it. I guess you could say a strict sense. Well, I've been on tafamidis now for almost a year and a half. I would say that I was at the most depleted point of my condition at the point when I started taking the tafamidis. The episodes of dyspnea were fairly frequent on occasions of anything more than moderate exertion. So if you know what it's like to ride trains in New York City, you'll know that coming up and down the subway involves climbing flights of stairs, and they can be longer or shorter flights of stairs. Before I was taking the medication, the instances of feeling strained, out of breath, short of breath, on infrequent occasions or even perhaps rare occasions, I would feel a tightness in my chest. As time has gone on since I've been taking the medication, those symptoms have either like been sharply have been mitigated to a great extent. I can walk up the stairs from the 125th Street station from, which is like 3 flights of stairs, right? If I take my time, I mean, I'm not setting any records climbing stairs, but it's not like a normal paced out kind of steady level of exertion to climb the stairs. Doesn't leave me like on street level, emerging from the stairwell, holding on to like the railing of the stair, wondering when my body's going to recover so that I can move forward without feeling dizzy, right? I don't have that anymore. I was at a point where, like when I was riding the trains, I wouldn't sit down because when I would get up, moving from a sitting position to a standing position, from a standing position to immediately climbing the stairs in the station made it even more difficult. So I had gotten into the habit of like just standing up on the train, never taking a seat. I'm finding that I have a lot more leeway in that regard. I would even say that my stamina on the stationary bike has increased.
One thing I want to add is that in terms of the speed of walking, not the distance, but the speed of walking is significantly slower. Just if we're walking together on the street, we can walk a long distance, but it has to be at a certain pace. Especially after I eat. And then after you eat, sometimes there is dyspnea after a meal. Not necessarily a large meal, but a meal of some sort. Yeah. And then it's a consideration is walking uphill.
Yeah, walking uphill, we take, as Patty's saying, we take our time going uphill. But all of that has gotten better in the last year since I've been on the medication and doing these other things. Even though there's like pipes that are like permanently sprung and like duct taped off, at least that plumbing is not getting any worse, right?
Tom would like to have less dyspnea. He would like to be able to be more physical without having to worry about that dyspnea, and in some case, reversal of the condition altogether, which seems to me to be, to a certain extent, possible in the future. So
maybe there could be some hope for some kind of additional medical breakthrough, which would allow for some remediation or repair or restoration of the tissues that were damaged.
I'm really delighted to introduce our next speaker, Bob, who is been very involved with amyloidosis support groups, and he has a family history, and I think a really important perspective. We often call people, bucket everybody as patients, but somebody who's been tested gene positive, we have to think about, who are you? Take it away.
Thank you, Isabel, and thank you to ARC and the FDA and the forum for the opportunity to speak today. As Isabel said, I'm Bob Gibson. I've been active in patient advocacy with the amyloidosis support groups for almost 7 years now, but I've been living under the shadow of hereditary amyloidosis for much more than that, almost 30 years at this point, ever since 1994, when my father was diagnosed with ATTR at the age of 58. At that point, his body was already ravaged by the disease, and despite a liver transplant, which he got within 4 months of his diagnosis, the amyloidosis continued to wear him down until it took his life in October of 2000. And so at that time, I wondered whether that was the fate that was in store for me or for my brothers and sisters. I'm now 3 years older than my father was when diagnosed, and in a different situation from his and from Mike's and Amy's and the other folks that you've heard speak so far. Although I inherited the T60A gene from my father, I've not yet developed any symptoms of amyloidosis. I'm not tested positive on PYP scans or MRI or any of the fat pad aspirates. But it doesn't necessarily mean I'm completely amyloid-free, but if the amyloid is there, it has not yet revealed its presence. So I'm classified as asymptomatic, or maybe just pre-symptomatic, or maybe just lucky, so far. So lucky or not, if you had asked me back in October of 2000 what a treatment benefit would be, I would have told you exactly what I'm saying now. And it's not to improve my symptoms, which I don't seem to have any, or recover any functions, which I've not yet lost, or they won't. The real benefit is not to lose them in the first place. And I think we've had this morning's discussion has really focused a lot around that topic, although obviously it's a complex subject. But preventing this terrible disease from occurring from the start is my objective. Do everything possible to avoid it, to keep it from infiltrating the heart and the nerves, thus the intestines or the kidneys, and slowly sabotaging their function. So preemptively stop it before it begins, because I know once amyloidosis gets its hooks in you, your life and the lives of those who love and care for you take a turn for the worse. So how much better off will we all be to avoid TTR amyloidosis in the first place? And again, after this morning's discussion, I don't view that as a rhetorical question at all. I know there are lots of people committed to doing just that. And we have treatments, but all those treatments, with one exception, can only be given after you begin suffering from the disease. And I think Mike mentioned that as well. No approved treatments are preventive. None have been investigated in trials for that promising use, and we need to change that. I'm the beneficiary of the one exception for preventive treatment. I've been snapping those 500 milligram tablets of diflunisal in half for 4 years, taking one in the morning, one at bedtime, in an attempt to forestall the disease from keep it from getting a toehold in my body. So as we know, diflunisal is the inexpensive and off-label alternative, or maybe progenitor, to tafamidis. So we, and I mean the team that I go to up at the Boston Medical Center, including Dr. Ruberg over there, we can't say for sure whether my daily diflunisal is preventing amyloidosis or delaying it, or really doing anything more useful than lowering inflammation and easing a bit of back pain. But it did raise my prealbumin, which, as we know, is a measure of the TTR protein circulating in my body. It raises up to target levels and has kept it there, which is encouraging. And I tolerate the dosage. It's an inexpensive generic, readily available at my pharmacy, and my local doctor can prescribe it. But obviously, diflunisal is not even close to being a magic bullet. It's not for everyone. Some people can't tolerate it, nor is it appropriate for people with certain conditions and diseases. And obviously, many doctors would not prescribe it for preventative use in the absence of any data suggesting that it actually works. But right now, it's the only option that I have. I do see TTR stabilizers like tafamidis and others on the horizon like acoramidis, both with good safety profiles, as obvious candidates for preventive use. It will be hard to get there, but that doesn't mean it's not worth the effort. So as we think about the stories that Mike and Amy and Tina and Thomas and the others that you're going to hear said today, I think, how much better would it be to avoid all of that pain and suffering and all that distress in the first place? For those of us who are threatened to follow in that dangerous and unhappy path, an ounce of prevention could be worth a lifetime of cure. Thank you.
Thank you, Bob. Very well said. We now have Hoss, who's joining us on Zoom. So if we can pull him up. Hi, Hoss. Thank you for joining us.
Glad to be here. Glad to be here. Thanks very much. First of all, I'd like to say thanks for the invitation from the consortium and this forum to speak on behalf of the patients. I think it's critically important that we don't lose sight of who the patients are and what this means to them. My name is Hoss Christensen. I'm 68 years old, and I'm a fourth-generation California farmer where we grow premium table vegetables. I have TTR with cardiomyopathy, and today, fortunately, it doesn't appear that there's any other major organs involved. My initial diagnosis was in March of 2019 by Dr. Watellis at Stanford Medical, and I later confirmed with a second opinion from Dr. Grogan at the Mayo Clinic. And I was very fortunate because at that time there was a tafamidis trial going on, and I was quickly enrolled in the trial and started the medicine within weeks of the diagnosis. I'd like to comment a little bit about what this disease means to me and my family. This disease is a very insidious disease, and that we all know that it shortens your life, but it also takes away a big chunk of, to me, what is living. I've lost sufficient amount of strength and balance that I can no longer saddle my horse. I can no longer ride to the foothills. And even if I could get on my horse, it's just too dangerous because I have persistent AFib, and I'm on blood thinners. The real loss there is not just for me, but I can't take my grandkids with me on that ride up to the foothills, something I've been doing the last 60 years. I can't take my kids to Disneyland or my grandkids to Disneyland because as you heard, I can't, most patients can't walk that far or that pace. It's just too much work for us, and it would slow down the whole group. It would be miserable for them. So that's another lost opportunity. The other big issue that most people don't think about is the fact that I really can't go anywhere where there are stairs. You've heard from other patients, I have this same problem. I can't walk up more than 5 or 6 stairs without needing a rest, or basically 2 handrails, which allows me to pull my body to the top. And when I get to the top of the stairs, I have to rest and figure out, okay, where am I? I'm a little dizzy, but I will keep going. So it's a different life for me now that I have this disease. Being on a farm, there are lots of things that I used to do that I now have to rely on my team to get done. And it all revolves for me around kind of general strength and general balance. But I still have a pretty good life because what tafamidis does is, and it's only, for me, it's just 1 capsule in the morning. And I thank Dr. Kelly and the entire team for creating this drug because it allows me to do these things. I can still take my grandkids to the creek and we can still look for frogs and pollywogs. I can still kidnap them out of school and take them to the baseball game. There's nothing more satisfying than having your wife or your grandma screeching the phone about, why are you at the baseball game? The kids are supposed to be in school. And I convinced her begrudgingly that baseball is just as important as school on certain days. And it's those kinds of small things that really add up. So again, I'm thankful for the researchers and the community to bring this drug to life for us. While tafamidis for sure isn't a cure, it definitely, my family and I are convinced, it definitely has slowed the progression of the disease. If we look at the rate of decline for the 2 years preceding to my diagnosis, there was considerable, I would say, monthly or quarterly decline in my ability to just basically function around the farm. Again, directly related to my strength and my balance. It was just disappearing, it seemed, at an alarming rate. So it was almost by accident. I was seeing my cardiologist because it turned out that I have persistent AFib. And his conclusion was, there's something more going on here. So he referred me to Stanford and Dr. Watellis, where I'm now getting the excellent care. So my ideal situation would be in the future, as pointed out by Dr. Gilmore, we'd have 1 set of drugs that kind of holds the disease at bay, while there's another drug that lets the body repair and removing the amyloid from, in my case, the heart. That's a much better world for me. Now, that may not exist for me, but I certainly want that opportunity for it to exist for my kids and for my grandkids, should they be impacted by this disease. I know there are trials going on with new antigens to try and do this, and I'm watching those closely because it doesn't seem like that's very far from reality. In the future trials, I think, are important. And I also think is related to a discussion earlier today. that the trials of multiple drugs trying to deal with the disease are equally important. I'd be willing to take those trials in the future, even though the risk benefits may be somewhat out of line with what I would normally do, but the potential benefit would be significant. So I think that for most patients, the issue with trials that we've all brought up, the fact that it takes time, it takes effort, and it takes energy to get there is really very, very important. So in closing, I'd like to again thank this group for the opportunity to speak. And I think it's critically important to always remember the patient. And I'm very encouraged to hear the discussions today because a lot of my concerns have already been brought up in the first half of the meeting, and I'm expecting more of my concerns will be addressed in the second half. So thank you, keep up the good work. We as patients are behind you 100%. And I think you'll find us a very cooperative group when you ask us for data or if you ask us for help. It's not very difficult from a Facebook social messaging standpoint to send out just a quick question. I get answers all the time from tens of people, sometimes hundreds of people who offer their opinion about a question that I have. So again, thank you very much.
Thank you, Hoss. And I think you just at the end touched on the importance of support that patients, particularly with rare diseases, can give each other and how important it is. We do have 3 more videos. It's so important to hear these stories.
My name is Arnold Shankup. The type of amyloidosis I have, I think it's called wildfire. Yeah, I have no idea. It's wild something. I'm sorry if I'm coughing. I just came in the hospital with pneumonia. I was seeing a physician at Morristown Memorial Hospital in New Jersey. And then he says, I don't think this is what you have. I think you may have, it's called amyloid that attached to your heart. And I want to see a physician we know at Presbyterian, Dr. Mora. I was on a study for Vindamax, and now I'm on a study for another protocol that is supposed to stop the amyloid from forming. And I have been on that study for over 2 years. So I would think it's like 4 or 5 years ago. Well, I would say presently, I go to the gym 3 times a week, but I have problems walking inclines and I get out of breath, but not really out of breath, but I got to stop walking. And I would like to change that, where I'm able to walk slight inclines or inclines, including steps, and not feel I climbed Mount Everest. I'm on a drug that's supposed to ease me, but some days I am very crampy and my stool is very hard and it's very annoying. That's one. The other thing, I don't know if amyloidosis affects your sleep, but I have problems staying asleep. I get up, I urinate, and then I go back to sleep. And there's always an issue with my diuretics. You know, how much to take, less to take, more to take as my weight changes. You know, my daily activities are not impeded because I'm an attorney. So most of the I'm sitting in an office. I go to the gym. I don't feel horrible. I could do a recumbent bike for almost 20 minutes without a problem of, you know, a breathing and stuff like that. So I'm pretty happy. It's keeping me alive. I don't have oxygen carrying around. I don't have a walker or a cane, so I could get around. So pretty good for someone 82. I like to be better, but it's okay. I think it's a new disease that's been around, but no one found out about it. And I was aware that I had carpal tunnel surgery, and Dr. Mauer said sometimes that's caused by amyloid. I have spinal stenosis, and that can be also caused by amyloid. I want to feel better. I want a drug that removes the amyloid from your body. That's what the important thing is. You have it, it's there. How do we remove it? So we gotta need research to get a drug to remove the damage, to remove the amyloid from the organs it attached to. That's the major thing. And that will help people now and in the future. Yeah, you can't cure it. You can treat it, but you're not curing it. And as you get older, it affects you more. That's for sure. And it should be treatable. It should be curable. That's what it should be. Thank you.
My name is Robert Chester. I was diagnosed with amyloidosis at the Mayo Clinic in September of 2020. I was told that I have ATTR-CA amyloidosis, which means cardiomyopathy, and the principal organ in my case is my heart, and a whole bunch of issues that apparently had been simmering for quite some time. I have had other involvements. I had prior to my diagnosis, in fact, many years prior, I had bilateral carpal tunnel release surgery, not once, but twice. I have also had told I had involvement of the amyloidosis in my shoulder joints. I have had both shoulder joints replaced in 2022. I had severe osteoarthritis, which I'm told was exacerbated by the amyloidosis. And let's see, I've also had CNS, central nervous system involvement, insofar as I have suffered from loss of libido and ED, very significant and abrupt over the last several years. And I also have involvement in my hands. I have trigger fingers in both hands. I neglected to mention at the outset that I also have had GI issues. With me, it's primarily constipation, which has been terrible. There have been times when it's been exceedingly painful when I've been constipated for an extended period of time. So it'd be wonderful to be rid of that. And also, as I have mentioned, the effects that it's had on my libido and on erectile dysfunction issues. That's also a very significant matter, because it does have consequences with respect to intimacy with my dear wife. So I would love to see some way of also dealing with that and alleviating those symptoms as well. So far, the urologist I've been consulting with has had me try a number of different potential treatments, none of which have been successful. A key benefit of treatment for me would be a reduction in the congestive heart failure symptoms that I experience on a daily basis, from the time I wake up until I go to bed at night. I used to be very active, and now I can't even climb a flight of stairs without having to stop and catch my breath. Oftentimes, if I bend over to tie my shoes, I have to stop and take a few minutes to recover from the shortness of breath that results from that simple activity. So a benefit to me would be a reduction or elimination of those symptoms, so that I could continue to be more active. I still walk every day when I can, when the weather permits. I have found that I can still walk fairly easily on a level surface. I can't walk at the pace that I have become accustomed to. And if there's any kind of incline involved, I have to stop and take it very slowly because of the shortness of breath, primarily from the congestive heart failure. In the development of any kind of treatment, clearly what would be primary would be to curtail the continuing progression of this disease, because it apparently continues to get worse, and that has been my experience as well. So we need to find some way to not just retard the progression as the current single FDA-approved treatment purports to do, but to actually stop it. And then if we had, a complete opportunity to choose anything at all, then to reverse it, to clear out some of these fibrils, particularly in the heart, that have caused the thickening of the heart wall, so that we can perhaps look forward to returning to the quality of life that most of us experienced prior to the onset of the consequences of amyloidosis. I think it's also very important in exploring potential treatments and perhaps even a cure, is to also consider the cost to the patient. I know for many of us, if not most of us, the cost is really burdensome, even with whatever insurance there may be available. For many, the copays are significant. Even for Medicare patients, such as myself, the cost of these medications, I understand that a great deal of money went into their research and development, but nevertheless, the cost to the patient, in many cases, from what I hear from others, is so prohibitive that there are patients who are unable to avail themselves of what is on them currently available by prescription, and have to resort to over-the-counter modalities that may or may not be effective. So I would hope that as new treatments are considered, that there's also consideration given to the ultimate patient cost. Thank you. Stay positive and test negative.
So with this, let me introduce Dr. Savitz. Please, thank you very much. Good afternoon, ladies and gentlemen. I am Dr. Barry Savitz, 81 years of age. I was diagnosed in 2012 and then was sent by a very observant cardiologist that worked with me at the hospital at Kingsborough Jewish. He was able to send me to the right place, Dr. Maurer in New York City at Columbia Presbyterian, for treatment and then inclusion in the protocol, which determined that tafamidis or Vindamax now is the drug for now to be used, with the understanding that it is not to cure, but it is to slow down the process to allow a much more functional existence. I've been advised by my cardiologist that I'm the longest living person that he knows on the medication, a little over 10 years, and I have amyloidosis of the heart. However, because our bodies are hardwired one organ to another in the need for keeping control of the fluids in the body, my kidneys are now on somewhat imperiled, but still functioning with a creatinine of 3.4. Well, I would like to see a drug that would eradicate the disease. It would stop it so that I could then learn to function optimally at that level, whatever level that happens to be, with knowing that it will get worse, of course, automatically, but that would be much slower demise, of slower downtrain. But meanwhile, I do want to express my appreciation again and again, as many times as possible, for the blessing that you've given me and that the medication has worked as well as it has. I think the next generation will be for medication that stops the disease, not just slows it down. I would like the my workers, my caretakers to know that they're giving me a gift. I appreciate their work. I appreciate those people who are coming into the diagnosis. They're somewhat lost. They feel, what is the future? What is the hope? Nobody knows the future for anything. And especially when I'm 89 years old, and when you get to be a certain age, you realize that you're old even if you're healthy. Basically, don't stop living. Just live lighter and learn to love it. And that's important.
And that concludes our videos. Very wise words from Barry to end on, and touching, very touching. I want to just spend a few minutes just asking a couple of questions to our panelists before we open up the session a bit. Amy, you talk about a wide array of symptoms. I wonder if you could just tell us sort of the 2 or 3 symptoms that are most burdensome to you.
Thinking quickly, I guess the latest symptoms that are in my way are ocular. The thought of not being able to drive, and I limit my driving at night, but losing my independence way too early is a huge fear. I'm also a theater director, so not being able to see well, not being able to hear well is a very frustrating experience. I've been through a bunch bigger struggles in terms of surgeries and things like that, but it's the day-to-day living that I think it's so very important and sometimes so very hard.
There's an aspect that we haven't really touched on about the psychological impact and for families. And one of the things I think we hear, often is that somebody has watched their grandparent die, has been a caretaker for their parent, and then has the disease. And I think the psychological impact of that on families is extraordinary. The other thing that we hear is how complex it is around the genetic testing and whether siblings decide to get tested or not. And do you have thoughts? I know, Bob, you come from a large family. I know you've connected with a lot of patients as well, and thoughts around that.
I do think there's a timing issue involved for when you would decide to get tested. I know that was in my case. I have a brother who also is a gene carrier. In fact, I have multiple members of the family who are gene carriers. He decided to get tested shortly after my dad was actually diagnosed, once we learned it was a genetic disease. And he tested positive. And so he's had this fear and this worry in his mind for years about developing the disease. And mind you, he also got tested before tafamidis was barely being explored, I believe, at that point. And the solution for all of us at that point would have been a liver transplant had we developed part of the disease. So he had that on his mind, but other members of my family did not, and we made that choice, and I made that choice to delay it, partly naively, just because I didn't necessarily understand as much as I do now about the development of the disease. But it was also because I was concerned for some real issues. Like, at that point, would it affect healthcare? Would I qualify for healthcare if I tested positive as a gene carrier? And this is, well before Obamacare. And secondly, life insurance, which I found out after I started being on treatment, I could not renew because amyloidosis is right out there on my health records. Not that I have it, but the fact that I am being preventively treated for it. So, there are considerations around that which I think are important and affected our various members of my families and the decisions that we made. But by the time we were all getting close to my father's age of onset, we all tested, and so that was probably the driving factor in that.
Do you have any comments on the decisions to genetically test, Mike?
Yes, I have a son who obviously went and got tested. I actually tested him under my name just as a precaution. But I have 5 nieces and nephews. My brother passed away, so it's impossible to test him. They refuse to get tested. I mean, they're all adults, 40s, 50s, you know what I mean? And I don't see the rationale to it, because if you test and you're negative, then there's no harm. If you test and you're positive, no matter what age you test, you need to go seek some medical attention for it. Your life insurance is not going to do you any good if you don't treat the disease you got, which is going to kill you earlier. You know what I mean? So I don't see any downside to not testing at any point. I mean, everybody can make their own decision, but if you test and you have the gene, then you open up the possibility, like if the imaging scans get approved and stuff, you can start getting these scans, and you can look. If you have the amyloid in your body long before any symptoms appear, then you can go on these other medications like tafamidis or Onpattro, whatever, and it can work start at a time before you have the damages, meaning then you're in pretty good shape from that point on. As it is now, we have to wait until we have symptoms, and when we have symptoms, well, our organs are already affected, so it helps, but it's not helping enough. Thanks.
Yeah, good point. It's interesting. I heard from everybody the issue of stairs and inclines. We do love our 6-minute walk, but it's not necessarily something that patients notice. And as somebody talked about, it was only his wife that observed him slowing as he walked. I am delighted to be able to introduce Robin Bent, who is Director of Patient-Focused Drug Development Program at CEDAR. And I'd love you to talk a little bit about what that program's done and, how the findings are used, and also how, as a community, when we've had such a change in the landscape from that first meeting, how can we share those findings with you?
Great, thank you. So it's really a pleasure to be here today. And so patient-focused drug development really started about almost exactly 10 years ago. And it really, when we first started patient-focused drug development, the goal was to use the information heard at patient-focused drug development meetings to help inform FDA's benefit-risk assessments. But I think as time has passed and we've had learnings, it certainly has helped inform FDA's benefit-risk assessments, but I think we've really learned that there's so much more value to these meetings, and I think your community has probably learned this as well, because these meetings are public, they're in the public domain, and so they're not just sharing the patient experience with FDA, they're sharing the patient experience with researchers and medical product developers and all the other people who are really critical to getting a drug through or a treatment through the therapeutic process, so that those people are also aware of what matters to patients. The PFDD meetings can give people new ideas for maybe targets for therapies. They can help to inform people about whether the endpoints that are being measured in the trials are endpoints that are really meaningful to patients. And so I think they do have great value, not just to FDA, certainly to FDA, but really so much more broadly. And so I think that, I think that the PFDD meeting that you all had many years ago certainly sounds like it's been a great starting point to kind of help move things forward as well.
Thank you. And as far as the second part, as far as as we've seen the patient experience change in the evolving landscape,
Mm-hmm.
is there some process to update that? And I'm sure you've had this in other communities as well.
So there are certainly different processes that can be followed depending on what your needs are, whether that's a listening session with FDA because you feel it's something that only FDA needs to be informed of. We have started to think about the idea of holding or allowing groups to hold second PFCD meetings if they feel like that's something that there's a dramatic difference in what matters to patients, and the landscape has changed so much that it's different. I mean, I saw the amazing survey results that you all put out, and I would encourage you to consider obviously sharing those, but also publishing those, because I think one of the things that we see so often is that patient groups do this amazing work and don't always publish it. And so when people are doing literature reviews or things, we can put things on the FDA website, but not everybody knows to come to the FDA website to look for things. So I do encourage, dissemination through kind of those traditional sources as well.
Excellent, thank you. And lastly, I'd love to invite Kristen to introduce yourself and just share if you have any additional thoughts.
Sure. So, can you hear me? My name is Kristen McCausland. I'm the Associate Director of Quantitative Research at Quality Metrics. I've been working in AL and ATTR amyloidosis for the past 8 years, trying to better understand the patient experience and also developing and validating patient-reported outcomes. I wanted to thank the patients for sharing their stories today. I interact with data elements all the time, and it's been so informative and helpful in our work, but it's really these touch points that really kind of remind us of why we're here, so thank you. In terms of the symptoms that were shared across the patient stories, I think they mirror what we've heard in other types of studies, both quantitative and qualitative research, that the most burdensome symptoms are often shortness of breath, GI symptoms, fatigue, balance. But what really struck me today when we kind of triangulate the sources of data that we heard, both the patient's stories and the survey research and really what I've come across in our own work, is that these aren't experiences that are characterized or defined by one or two really burdensome symptoms. This is a really complex condition, and really kind of looking at the breadth and the depth of the symptoms experienced, we might really need to think about this holistically. I think it's really critical to look at the individual level symptoms for a variety of reasons. I think it informs unmet need for clinical purposes. It could highlight where we could provide extra services or supportive symptom management. It can inform the drug developers for where we can target new therapeutics. But at the end of the day, they're not happening in isolation. And I think when we talk to patients, they talk about the impacts, the health-related quality of life impacts, the functioning. And I think that those are really going to be important considerations when it comes to defining meaningful treatment benefit. And so I think that that's really the take-home message I heard.
I think that's a great comment to finish this on, that in a multi-systemic disease, we need to look at patients holistically, and so much of the medical system is set up against that, from the way regulatory divisions are set up to the way siloed professions of specialties. And for patients, it's a combination of symptoms that become overwhelmingly burdensome, and we need to do a better job at really helping patients for their disease and not having patients, a lot of patients we have an amazing physician patient who spoke recently about, he spends 70% of his time managing all his different doctors for each symptom. It is one disease that we need to do better at looking after holistically. So thank you all very much, and I'm delighted to hand over to Matt now.
Thanks. It's always, I have to say, inspiring to hear the stories, and if that doesn't move you, you need to find a new job, I always tell the junior attending. So I appreciate everyone by video and in person and by Zoom for sharing those insights. Maybe I can ask some of my colleagues. I think, hearing that, we don't do a very good job at, addressing the multi-systemic nature of the disease. So I don't know if people have suggestions, but what can we do better in this particular regard? Any ideas are welcome.
I can speak up, Matt. One thing that dominates some of our patients' symptoms in the office is pain. Chronic pain, as we know, is a life changer, and immobility. So often these spinal stenosis issues, these orthopedic issues, we don't think about, putting somebody on treatment until they manifest in the heart, but that can impact someone's life more than heart failure. If you can't walk because you're in pain, or you have neuropathy, or can't go grocery shopping, I was really moved by all these things, that just basic things we take for granted that patients can't do. And honestly, half the visits are about pain and immobility, and less so about shortness of breath. And obviously we don't capture those things, but they're super important to our patients.
I don't know, Martha or Julian, things to add in your experience that you're seeing?
I think, the patients, thank you to all of them. They've highlighted so much the challenges. I think as far as the multidisciplinary approach, we try, but an unfortunate limitation of our healthcare systems, probably pretty much worldwide, especially for the physician, is the amount of time with the patient. And that's where, as we expand more multidisciplinary teams, I think we are better able to address the patient's needs. And that's where we rely so much on the support groups and ARC and others to help us with that. But it's not that people are trying to be bad doctors. There are just realities that make it very, very challenging. But having said that, I think the key is to really have a network to support patients. And once we have that, and when we have it to some degrees, but it's fragmented right now. So I think that's the best way to have a more holistic approach to our patients, and they usually find it very helpful.
A question from the audience here, please.
Hi, Matt. It's Deb Bodeker from Mackenzie's Mission in the Amyloidosis Speakers Bureau. We focused on raising awareness amongst the training physicians, so the medical schools, the residency programs across the country. And now after 4 years and 250 presentations to over 12,000 students and residents, it's fascinating to get some feedback and data points. And I'm reacting to your comment and your question, Matt, about multisystemic diseases and how they're taught in medical schools in the training, where so often our patient educators say, it took my doctor, my fourth doctor that I saw, took them stepping back and looking across my body, across the systems, to try to connect those dots. And I just wonder if, there's a lot of blinders when you're in specialties. How can we help the medical education to help them think more broadly when they come across more complex diseases such as amyloidosis? I don't know the answer, but it seems like we run into that a lot, given the questions that we get from these medical students and even residents who are working on developing their diagnostic skills. Food for thought.
Yeah, I don't have a solution. I would say that Mackenzie's Mission has done a fantastic job at bringing amyloid awareness to a whole host of new providers. I have to say my own personal struggle at our center is that I think a lot of the, if you will, extra cardiac manifestations, I don't think we have a lot of expertise that's present. So, I don't have a bunch of orthopedists who are actually interested in, studying, learning, or managing patients who have, as Matt says, chronic pain and orthopedic issues that are preeminent. We heard a lot about gastrointestinal symptoms, and I think we probably could do a better job at trying to bring other subspecialists into the fold in some regard. That's one hot button item. Julian?
Yeah, I just wanted to mention that it was extraordinarily humbling hearing from all the patients, and I think there were a few messages that recurred. I think, and one was prevention, and I think that prevention is something that we obviously all think about, but are we doing enough about prevention? The second, obviously, was early diagnosis, which we've talked a lot about today and is improving, but there's obviously room for a lot of further improvement. Another recurring theme, which related to Kristen's talk as well, was the fact that 50% of patients don't know whether the treatment that they're receiving is impacting their disease. I mean, that's a staggering statistic. And we heard from a number of patients about the desire, obviously, to get improvement rather than just stability of a disease which is massively impacting their lives. So I think there's so many themes there that we as a community need to focus on. And just to reiterate everything that my colleagues have said, I think the multi-system nature of the disease was so apparent, wasn't it, from all those 9 different talks that we had, and there are so many aspects, many of which we've touched on already, that we as clinicians just don't focus on and need to try and improve our focus on.
Yeah, I completely agree. Maybe I just ask the patients, is there something you think we as providers could do better in engaging you in your care? Tracking the disease better, getting the patient's voice louder in the office. I'm just, suggestions that you think would be either complicated or simple that might, or you've seen really affected how you were cared for. Haas, you look like you want to say something. Go ahead, Haas.
Yeah, so Matt, I think one of the things that once you've actually been diagnosed by, let's say in my case, the cardiologist, it's been really useful to have a list of, here are other symptoms that may be related to this disease, because the cardiologist may not be able to go down through all and say, hey, look, there's a potential for kidney issues here. Are you having GI issues? Do you have an ED problem? All these other things, if they're kind of, I hate to simplify it, but simply like a checklist that you could go through and say, yeah, I've got some of this. Yeah, I've had, in my case, I had carpal tunnel releases done, right? I have severe osteoarthritis. It's all related. I've also had a torn bicep. These are all things that, now I know are related. But when I was first diagnosed, without doing some homework, I didn't know those could all be related. So providing just that simple, I'll say broader horizon of potential issues to the patient once it's actually been diagnosed, I think would be extremely helpful.
Any other thoughts or comments?
I have just one or a couple of comments, but I think often patients find it very hard to raise symptoms that might be socially difficult. So erectile dysfunction is a very good one that we'll often hear from patients, but they won't have told their physician. I think the other one that patients have, and we've spoken about this, extreme anxiety about affording treatment. So even when they are part of a program that is covering their co-pays or they're on whatever it might be, the fear that that won't go on and that they'd have to renew. And I think we really overlook how much of a burden that financial toxicity and anxiety is for patients.
Yeah, I would agree. I mean, patients are administered, as you know, probably tafamidis at a monthly basis, and comes to the 4 or 5 days before the next box is coming, and they wonder, is it actually going to come? The stress around renewals on a yearly basis. If you're a large program, I mean, we'll talk about this afternoon, but access is incredibly complicated, and without specialty pharmacies and support staff, there would be no way that programs that are caring for hundreds of people could actually manage this. I just wanted to return to the insights you were providing about genetic testing for, at-risk family members. Maybe you guys can comment a little further. Is there a better approach that you think we could be utilizing in communicating this information?
I think if doctors were to talk to the patients and, explain it to them in more detail, and, like family members probably tend to get a little emotional if people aren't listening to them, you know what I mean? But if a doctor could break down exactly, how this is going to help them, and, in the future, I think, that would probably persuade more people than, just arguing over the kitchen table about it.
let me comment on that too. I think timing is very important, right? So, we know with many of these varieties and variants that they affect people at different life stages. So, in the case of the T60, which we know tends to affect people 50 and older, do you really want someone to be tested at age 25 or 30? It doesn't make much sense to me to have that. And I can see the hesitancy for someone in that age range, despite knowing about the disease because of a parent or another relative who might have it, just being not certain to take or not wanting to take that step. And it seems like it's reasonable not to necessarily take that step. You have other things that you're considering at that stage in life. But then if timing is an issue, then the issue is at what point are we communicating with people who might have the disease or might be genetic carriers? How and when are we communicating them that now is an optimal or at least a good time for them to get tested? I'm not sure the answer. I think that's going to change with different family situations, but I think it's an important one to try and consider as we broach that to potential carriers or potential patients.
I've had some interesting experiences that kind of flip some of these things on its head, because I went to, at some point, a neuropathy specialist. I mean, somebody who, was considered one of the top experts in the country about this. And, I was basically, I had been questioned about my family history. Do you know anybody in your family who has had anything like this? No, no. And I also, one of my misdiagnoses was Sjogren's syndrome. And I had dry eyes and dry mouth with, then they said with neurological symptoms or neurological complications. And I tested negative for Sjogren's syndrome. I had a positive ANA, negative for Sjogren's syndrome, but they said, well, you're probably going to test positive within the next 2 years. I went to a Sjogren's specialist, both where I was living and where I was moving to, and was told, this was not Sjogren's, you need to go back to a neurologist. Went back to the neurologist. He said, well, you told me this didn't run in your family, so there are 4 things that cause neuropathy. I've tested you for 3 of them. I'll test you for the 4th, but most likely it's going to come up negative and we're going to be treating symptoms. Of course, it didn't come up negative, and it came up with this extremely rare variant, and we're off to the races. Years later with lots of damage done, but there was no treatment before that anyway. So anyway, it's interesting, the blinders question, as well as, there weren't names for things when people died of heart issues or other things that may or may not have been related. You could find anybody in your family who may have had something, a couple of generations ago. Anyway, that's been my experience. And of course, living with my kids, trying to figure out what to do has been a really rough thing. And psychologically, they were very young, especially for kids of somebody with amyloidosis. So they grew up, with suicide ideation and worse because they're watching their mother, waste away. And, then that gets so much worse to think about if they were to get tested at 18 and have a psychological noose around their neck until or if they ever develop symptoms. And so it's a very complex situation.
Yeah, that's my sense, is that every family is very individualized, and the discussions are nuanced. I don't know. I always try to be upbeat about this and think that, we've made a lot of progress, and for the next generation, it will be much better. But I can't imagine watching your mom or your dad get sick and then think that could happen to you. Martha, you have your hand up for a while. Sorry.
I was so glad to hear the highlights of the psychology of genetic testing and why there is resistance, because I think healthcare providers sometimes have this, oh, it's so important that your family members be screened, yet we've heard the psychological downsides. And we have had complementary programs that have been offered, but I think it's very important as a community to highlight that genetic counseling needs to be done before the genetic testing. Sometimes it's been genetic counseling after the genetic testing, but because the psychological impacts and all the impacts are so important, but also now with virtual visits, I think it's important that it's a genetic counselor who really understands hereditary TTR amyloidosis, not just a generic genetic counselor who tells you something about life insurance and medical insurance. There are some variants, they're relatively rare, but that can present very early in life. So for some people, earlier testing will be important. So I just think it's so important. And some of my hereditary patients have told me the hardest thing with their whole diagnosis was to explain to their family members that they might have this condition. Harder than getting a left ventricular assist device and all kinds of other things. That's been the hardest thing. So I'm glad that was highlighted, but I think we really need to have expert genetic counselors interfacing with the family members.
Thanks. And Care, maybe the last comment before lunch.
Thank you, Matt, and thank you to the patients for sharing their stories and hearing all this. I'm just wondering, as a field, as a group of people invested in this disease, is it time to start advocating for perhaps a treatment approach that emulates heart transplant or pulmonary hypertension, where it's an expectation for a treatment center to have a minimum standard of people involved in the care of these patients? And maybe it's not as expensive, but maybe a patient liaison, maybe a psychologist that's involved, pain management, because it seems like this is our struggle. But maybe if I wouldn't want to say the word mandate, but a center of excellence expectation or something of that sort for this is how you do it, blueprint.
Yeah, I mean, sometimes mandates like that are very helpful for us to advocate at hospital administration levels and so forth. Reflecting on the idea that a 340B pharmacy was started decades ago to provide HIV therapies to people who are underserved and to provide resources for individuals who are newly diagnosed and social workers. So maybe we could, figure out some way to claw back some of those funds to support, I think, the patients in a better manner. Well, thank you all for a very dynamic discussion this morning. Enjoy lunch, and we'll come back at 1:30. Thanks.